son más que un grupo de investigadores, son un mecanismo para poder lograr la mejor cantidad de información, de mejor calidad y de mayor impacto para lograr publicaciones de alto nivel internacional. Los que no estamos en institutos o en centros de investigación y trabajamos como investigadores solitarios un poco, tenemos que luchar entonces diariamente con lograr el mejor mecanismo, y uno de los mecanismos que usamos los que trabajamos en ese otro ámbito es, tal como lo usan los institutos y centros de investigación y universidades mayores, es generar colaboración con grupos locales y con grupos internacionales. Es así entonces como implementamos nuestras ideas y es así como aumentamos la calidad de nuestros resultados. Entonces, se parte por definir un journal y buscamos el mejor journal que se adapte a nuestros resultados, y ahí dice algunas tips que les muestro, por ejemplo, cuál es el mejor journal o el mejor high quality journal para nuestro set de resultados. Y en este caso decimos es importante conocer qué journal está publicando hoy día o va a publicar datos, pero en base no solamente a lo que se publica normalmente en ese journal, sino quién va a leer ese trabajo que ustedes están preparando para enviar, ¿no? Entonces, es muy importante centrar nuestros trabajos en el journal y la calidad y además el ánimo final de ese journal para la gente que lo va a leer. Siempre uno busca un journal de alto impacto o de alto reconocimiento científico, pero puede que ese journal no represente realmente la audiencia que va a leer el trabajo de ustedes. También hay que preocuparse, por ejemplo, que los journals tengan una calidad de reviews o de revisiones que hagan los revisores que uno considera aceptable, que uno considera que se quede tranquilo con los comentarios. Muchas veces uno discute cuando llegan los comentarios y dice, este señor no leyó el trabajo o simplemente no sabe de qué estoy hablando yo en mi texto. Y eso puede ser muy verdad, pero también es verdad que nosotros eventualmente recibimos críticas de revisores, de aquellos que se han esmerado para hacer su revisión, que no nos gusta, porque no nos gusta recibir críticas, así de simple. Es difícil que uno acepte a buenas y primeras que le digan que su sección tanto de su manuscrito no tiene sentido y no se incorpora al texto general de su trabajo. Uno sufre con eso, pero tiene que aprender a aceptar justamente eso, y también hay que elegir entonces un journal pensando en que los revisores que van a estar allí quizás ya tengan un buen background y las revisiones hayan sido hechas de otros trabajos, hayan sido realizadas en el contexto que uno quisiera en realidad como un feedback positivo más que negativo de los revisores. Por supuesto, ver si este journal está bien localizado en el mundo científico, si es un journal respetado. Hay muchos factores, pero los principales son los índices que se utilizan para calificar un journal. Y algo muy importante, darse cuenta o intentar identificar o ver las listas que están publicadas de journals predatory, ¿cómo se dice? Predatory, no sé, journals. Estos journals son muy atractivos porque muchos de ellos tienen un factor de 7, 8, 9 incluso, asociados a $3,000, $4,000, $5,000. Entonces es muy importante identificar estos journals y darse cuenta que no es solamente una oportunidad de publicación, que lo es, pero al mismo tiempo en algunos comités, y aquí hay profesores con mucha experiencia que seguramente han estado en muchos comités, en algunos comités hoy día se está evaluando si los currículums están representados por journal predatory o no. Entonces una persona puede tener, no sé, 200 papers y la mitad de ellos son los que se reconocen como predatory journal. Eso a un individuo más viejo como nosotros, o alguno de nosotros, puede que no nos afecte mucho, pero una carrera joven de un individuo que está partiendo, quizás un currículum de 10 con 8 predatory y 2 no predatory, quizás no sea muy aceptable. Así que ojo con eso. Creo que la calidad del journal es muy, muy importante al momento de identificar. Simplemente, ¿cómo elijo el journal? ¿Ya? Hay que ver, primero que todo, los resultados que uno tiene. Y en base a los resultados, elegir el journal. Mucha gente procede eligiendo un journal y entonces hace resultados o hace experimentos para ese journal. Pero la verdad es que uno tiene un objetivo mayor, que es la tesis, que es un proyecto, o que es simplemente investigación que está haciendo por mucho tiempo. Entonces, ver el significado de los descubrimientos que tiene o los resultados que tiene y elegir entonces un journal. Fijarse en quién va a leer tu trabajo y fijarse cómo o qué camino sigo para elegir tal o cual journal. O sea, cómo lo hago para elegir un journal. ¿Okay? Eso es algo crítico en el buen camino de la publicación. Ahora, también es importante ver, por ejemplo, que las suscripciones a los journals es hoy día una materia de discusión permanente. Y las suscripciones a los journals es algo que nosotros muchas veces no entendemos, porque algunos de estos journals son absolutamente online. Entonces uno dice, ¿por qué tanto costo para publicar un documento online? Bueno, igual hay un maqueteo, hay un arriendo de plataformas online, que no sé si, yo incluso dudo muchísimo, que un maqueteo de un paper de 5 o 6 páginas con 6 o 7 figuras vaya a costar $3.500, que es el promedio de lo que cobran en revistas comunes y corrientes. Entonces, muy importante elegir un journal que tenga un costo, si es que están decididos a pagar por un costo, que sea un costo aceptable y razonable en términos de los financiamientos que uno puede tener. Hay journals que parten de $300 por publicación, pero eso viene acompañado de una segunda página que dice, si usted pone la figura en horizontal va a costar otros 200, y si lo pone en vertical, no sé, 350, y así, y al final suma como cualquier boleta, digamos. Que sea un journal de open access es muy importante, ¿cierto? Ustedes entienden eso. Entonces, claro, las editoriales han buscado el camino de hacer la necesidad del investigador open access y al mismo tiempo ganar un poco, sacar un poco de profit de esa necesidad de ese investigador. Esto es marketing al final. Yo vendo papas fritas con mucha sal y las vendo más caras. Papas fritas con poca sal las vendo más baratas, porque a la gente le gusta la sal, así de simple. Lamentablemente, a propósito de obesidad, eso está muy mal, así que no es una propaganda comprar papas fritas con sal, por si acaso. Hay unas papas fritas que son hoy día con muy bajo nivel de sodio. Y que el journal sea realmente un journal que uno reconozca como legítimo, como eventualmente en el camino y en la lucha por publicar y todo. El predatory open access publishing es muy importante porque la gente dice, bueno, esto va a ser open access, tu paper está aceptado, es un open access, tiene costo que es tal cual, y la gente decide si lo paga o no lo paga. Uno tiene ahí un momento en el proceso de envío de paper si acepta o no pagar el open access. Y si no lo acepta, cae en que la institución o de donde tenga su afiliación eventualmente tenga un acuerdo con la editorial y entonces no paga. Muy bien, si la universidad lo hizo, el instituto lo hizo y está pagando por tu trabajo, mucha gente lo acepta y está muy bien y aprovecha de publicar. Pero cuidado con los open access predatorios, porque podemos tener esos mismos convenios con universidades o institutos, pero aún así siendo todavía un predatory journal. O sea, la universidad, muchas veces universidades mayores, grandes, que uno llama los monstruos de universidad, no se dan cuenta algunas personas que están pagando $3.000, $4.000 por un paper que en realidad podría costar por publicación máximo $500. Pero como se hizo convenios, entonces se sigue funcionando sin mucho cuidado. Ocurre, por ejemplo, en universidades, ocurría en universidades, a mí me pasó, descubrí una situación y dije, ¿por qué está ocurriendo esto? Bueno, una universidad decía, nosotros compramos los pasajes suyos para ir a Argentina, por decir algo, y no se preocupe usted. Entonces uno dice, ya el pasaje cuesta $1.000, ya, por decir algo. Pero después llegaba una boleta a la facultad donde el pasaje costó $3.500. No, y tenemos un acuerdo con la aerolínea. ¿Okay? Entonces, ojo con los acuerdos de Open Access Publishing de distintas instituciones con predatory journals. Después de todo, nuestro interés es la importancia del journal, es cuáles son sus índices, básicamente. Y hoy día trabajamos lamentablemente con el impact factor. El impact factor es algo que nos mueve plenamente. O sea, yo discuto aquí en la sala y digo, ¿qué impact factor quisiera usted publicar? Por supuesto queremos el mayor impact factor. Y es la herramienta que se usa tanto para promoción académica como para asignación de recursos para proyectos de investigación o becas para estudiantes, etc. Así que es algo que tenemos que considerar. Pero también tenemos que pensar que hay algunas deficiencias de lo que significa el impact factor, que hoy día la gente está considerando muchísimo y eventualmente se está moviendo a otras consideraciones para determinar si su publicación está en un journal de alto impacto o no. Y justamente eso es. El impact factor no es exactamente qué tan impactante fue tu trabajo para la comunidad científica, sino que es cuántas veces lo citaron en un determinado periodo de tiempo, en términos de todas las citaciones. Entonces es una figura, un número, pero no es realmente una valoración del impacto de tu trabajo. Y ustedes habrán leído muchos estudios ya en revistas pequeñas que llamamos, ¿cierto?, que tienen un índice de citación de 5, 10, 15 años, más. O sea, ha pasado 10, 15 años y la gente sigue citando. Esos trabajos no necesariamente están en las revistas de impact factor sobre 10, sobre 15. ¿No? Así que es muy importante fijarse en el impact factor, pero es más importante fijarse en los resultados que uno tiene y cómo entrega el mensaje. Aquí hay, por ejemplo, un gráfico que muestra en las barras grises un aumento del impact factor en el tiempo, no sé de qué año, 90 y algo, hasta el 21. Sin embargo, eso es el global del impact factor, cómo ha ido aumentando la necesidad de un grupo determinado de revistas en la comunidad científica y cómo ha ido aumentando el impact factor, por muchas razones. El open access ha ayudado con un costo asociado, ¿cierto? Ya lo he estado mencionando hace rato, pero hay revistas que mantienen un impact factor por todo este periodo de tiempo, del 97 al 2021. Por ejemplo, aquí está obviamente de la Physiological Society, está el Journal of Physiology mostrando en el área de fisiología, en este periodo de tiempo en que se mantienen estos puntos verdes a lo largo del tiempo. Pero no va subiendo eventualmente todos los años como ocurre con el impact factor global. ¿Ya? Entonces hay algo aquí interesante, que si yo me posiciono en 1997 y luego en el 2021 y yo estoy en ese camino, estoy feliz porque aumenté mi impact factor, pero varios puntos importantes. Pero si yo publiqué en el 97 en el Journal of Physiology y publico en el 21 en el Journal of Physiology, el impact factor de la revista en general sigue siendo estable. Pero ¿qué es más importante? ¿Que subió, que bajó o que se mantuvo estable? Esa es la pregunta que tenemos que hacernos como investigadores, básicamente. ¿Dónde compro yo un vehículo? Compro un vehículo nuevo donde sé que la empresa tiene reputación por largo tiempo. No compro un vehículo nuevo en una empresa que surgió ayer y que vendió 500 autos en un día. Es un chispazo de un día, una fotografía de un día. Si uno mira otra área, en el mismo, en la misma sección de fisiología hay áreas que pueden ir bajando su impact factor en el año levemente, pero que siguen siendo altamente importantes. ¿Quién diría que un journal de fisiología y neurofisiología no va a ser importante? ¿No? Entonces, ojo con maravillarse con las publicaciones que tienen o que muestran simplemente un aumento del impact factor cada año. El índice inmediato o de inmediatez es algo que se está utilizando hoy día para las revistas. La citación o la vida media de la citación es muy importante. Uno encuentra incluso en los perfiles de uno mismo, de Google o de otros, encuentra la vida media de tus publicaciones, de la citación de tus publicaciones, y eso es algo que uno no conocía hace poco, hace ya 5 o 8 años quizás empezamos a utilizarlo, y en algunos currículos ahora la gente incluye la citación media de sus trabajos. Después hay factores nuevos como el aging factor, que está iniciándose recién en la literatura y todavía no se entiende mucho cómo va a afectar a las publicaciones. Pero en general, ¿cómo preparas tu trabajo para una buena publicación? El proceso general y todos los elementos incluyen primero la autoría. Y aquí caemos en algunos vicios importantes en que la decisión de la lista de autores parece ser una herramienta que a veces se escapa de los investigadores principales o los estudiantes involucrados directamente en el trabajo. Y caemos en incluir autores en publicaciones que eventualmente no hicieron nada, pero hoy tenemos un compromiso académico, personal, social con esas personas. Y eso es algo que eventualmente tenemos que ir sacando de nuestras actividades si es que lo hemos hecho en algún lugar. Es importante decidir entonces la secuencia de autores, la calidad del trabajo realizado por cada uno de los autores y dar una autoría a quienes lo correspondan realmente. Los criterios son, bueno, que una persona va a ser autor si ha generado ideas de la concepción original o en el desarrollo y diseño del trabajo, si ha analizado y ha interpretado los datos como corresponde, si ha participado realmente en eso. Hay gente que uno le envía, por ejemplo, una tabla con todo el set de resultados y la respuesta es: yo cambiaría el celeste de la línea 2 por un amarillo. Y eso es autoría para algunos individuos. Entonces, hay que tener cuidado, sobre todo la gente joven, sin duda, que está entrando en esto, cómo va a proyectar su respeto y su autorrespeto por su propio trabajo, ¿no? Es muy difícil, sin duda, para grupos mayores en que un estudiante recién iniciando pueda alzar la voz y decir: yo no quiero que tal persona vaya, porque se genera un conflicto en nuestra sociedad. No solo en Chile, afuera también ocurre. Pero eso tiene que cambiar, así de simple. Y es por eso que han surgido reglamentos publicados en Nature, en Science, diciendo: estos son los requerimientos para ser autor principal, autor secundario, intermedio, etc. Y en eso tenemos que basarnos, porque ese fue el consenso internacional propuesto por un grupo mayor de científicos. Así que hay que definir muy bien cuáles son los criterios de autoría, y eso no están en nuestras manos nomás, sino que hay documentos con reglamento específico. Incluso cada journal tiene algún tipo de reglamento específico. ¿Dónde comenzamos a escribir un proyecto? Bueno, hay elementos, obviamente se tienen que tomar decisiones, como por ejemplo, quiénes van a participar del trabajo, ¿ya? Eso puede ir cambiando en el camino, hay que elegir y hacer una buena revisión de bibliografía, es como el punto de inicio. Ustedes sabrán, los profes siempre dicen: ya, hagamos esto, pero revísate este tema en 2, 3, 5 días o quizás un par de semanas. Algunos estudiantes dicen: pues dame un mes. Y uno dice: sí, claro, un mes en ciencias son como 6 meses en realidad. Así que un día podría ser que ya es como un mes. Pero ese tipo de cosas hay que hacerlo, hay que hacer revisión. Y aquí sugerencias solamente, estas son solo sugerencias, y que dice: escriba sus métodos a medida que vaya terminando sus experimentos, porque usted hace un experimento, tiene conciencia de que lo hizo de la mejor forma posible, y escriba los métodos, porque si los deja para 6 meses después se le va a olvidar algunos detalles de los métodos. Y a muchos no les pasa, pero a muchos sí nos pasa, que se nos olvidan algunos pasos específicos de metodología. En efecto, cuando uno comienza a escribir resultados, cuando ustedes están trabajando en sus datos y empiezan a escribir resultados, lo más lógico que pase es que se encuentren con un gráfico que le falta el panel D. Tienen A, B, C, y la pregunta que ustedes mismos tienen no está hecha experimentalmente. Entonces, a medida que van viendo sus resultados, van a poder ver qué es lo que les está faltando. Y una cosa importante que una vez se deja de lado, y que los estudiantes a veces dicen: no, a mí no me interesa eso todavía. Bueno, error, debería interesarles desde el primer segundo que entran al área académica, científica, y que es considerar tanto las consideraciones prácticas, o sea, cómo hago mi trabajo, ¿ya? Y las consideraciones que llama aquí en comillas políticas. Las consideraciones políticas son amplias de definir, pero esencialmente nuestro trabajo es cómo hago esto sin afectar a otros, sino que simplemente beneficiar a otros. Y esto es muy interesante, porque a veces hay críticas en papers de gente muy importante o gente más joven que está partiendo, en el que critican trabajos de otros grupos y lo critican malamente. El trabajo de un editor bueno, va a ser: ¿sabe qué? Su texto está muy bueno, pero no tiene por qué ser peyorativo con este grupo de autores que no hicieron lo que usted pensó que era lo correcto. Entonces hay que ser cuidadoso, hay que ser muy cuidadoso con lo que uno escribe, tanto en todos lados, en métodos, en todos lados del manuscrito. Entonces, los elementos esenciales que se proponen, esto es una definición de la Physiological Society principalmente, son preguntarse por qué se hizo el estudio para poder escribir algo. Antes que todo, y las discusiones con sus profesores ya es simplemente dice: mira, tengo este resultado, profe. Entonces uno dice: ya, partamos por algo. ¿Por qué hiciste el experimento? Y si no hay respuesta a eso, el resultado casi no tiene significado, ¿me entiendes? Entonces uno tiene que saber por qué hizo el experimento, que es necesario y que es absolutamente normal si uno dice: bueno, voy a hacer tal experimento porque tengo que resolver tal pregunta. Pero hay muchos casos en que llega alguien con un número a la mesa y no sabe por qué hizo el estudio. Eso es terrible, ¿no? Después, ¿cómo se hizo el estudio? Hay que definir los métodos, sin duda, eso parece ser un poco más fácil. ¿Cuáles son los resultados que encontraste? Y cuál es el significado inmediato de ese resultado que encontraste, ¿ya? Entonces, en el contexto de la pregunta esencial de tu investigación, tienes que partir por qué voy a hacer el experimento, hacer lo que corresponde y entonces interpretar esos resultados. Un ejercicio importante que se ha hecho y que yo lo aprendí de mis tutores también es que yo tengo un resultado y lo trato de interpretar en su totalidad como el resultado. Un gráfico, y digo, ya, ¿qué significa? Este es mi mundo entero, es mi gráfico. Interpreto mi gráfico. Eso no significa que mañana vaya a ser exactamente la misma interpretación, porque mañana vas a tener más resultados. Y pasado mañana vas a leer una literatura en que se informa algo diferente a lo que tú estás planteando. Entonces tu respuesta al significado de tu resultado va a ir cambiando permanentemente. Pero un buen ejercicio es interpretar tu resultado como si ese fuera el mundo completo que estás viviendo. Los métodos, y aquí voy a ir más rápido, por supuesto ustedes ya saben, hay que ser preciso, hay que describir. Hay cosas de forma también, por ejemplo, cuando dice identificar los modelos y los reactivos usados. Uno puede encontrar lo trivial esto de decir, bueno, usé cloruro de sodio de sigma. ¿Qué importancia puede tener eso? ¿Cierto? Esa es la pregunta que uno puede hacerse. Pero un revisor astuto va a decir, bueno, usaste cloruro de sodio de otra empresa, ¿está purificado? ¿Está realmente puro como tú lo quieres? Entonces necesitamos saber, los que queremos repetir tu experimento, con qué reactivo hiciste tu experimento. Ni hablar en términos de anticuerpos y otros reactivos mucho más específicos. Así que hay que ser muy cuidadosos en los métodos para poder lograr un buen resultado. Los resultados, simplemente describir y describir con la intención de dar respuesta a tu pregunta de ese grupo de resultados, para publicar, me refiero. Y aquí hay una parte que dice, recuerda que al describir estos resultados en este paper, no estás describiendo tu tesis completa. Normalmente las tesis son 3, 4, 5 papers. Entonces, la intención del que escribe es poner la idea de su tesis en un paper. No, el paper es el paper, es el grupo de 5, 6, 7 figuras. Ese es. Entonces, tienes que describir solamente los resultados para tu publicación, sin perder de idea, por supuesto, en lo que significó tu tesis, pero el estudio es el estudio. Y ahí abajo decía, cuéntanos una historia a la revista. La historia, el revisor y los revisores y el editor quiere escuchar tu historia. Está dispuesto a escucharla, y qué mejor que contarle la historia. Un muy buen ejercicio, que seguramente muchos lo han hecho, es contarle al amigo o a la amiga, esto es lo que estoy haciendo. Y ahí, y yo he notado cuando estamos en reuniones sociales y todo, alguien le cuenta a otro y yo lo escucho, porque no me invitaron a la discusión, pero uno lo escucha y dice, sí, exactamente eso es lo que está haciendo, pero se lo dijo con manzanas y peras a ese individuo. Y después viene a hablar con uno y uno dice, a ver, mira, dime qué estás haciendo ahora con este experimento. Y empiezan las palabras rebuscadas, no, es que estoy experimentando con la disociación del... No, momento, ¿qué estás haciendo? ¿Entiendes? Dime lo que estás haciendo, tal como le dijiste a tu amigo hace 5 minutos atrás. Y eso cuesta muchísimo, cuesta porque el alumno cree que si no lo dice con palabras bonitas, el profesor no le va a gustar. Al contrario, yo creo que aquí está el profesor Juez y muchos otros profesores que quieren escuchar en lo más simple tu resultado y después vemos lo específico. Pero ¿cuál es tu idea? Saber que lo sabes, eso es importante. La discusión, ni hablar, la discusión, destacar las cosas más importantes. Y algo que a veces uno encuentra al revisar papers es que no se destaca o no se destaca claramente cuál es la respuesta a la pregunta inicial que bien hiciste en la introducción. En la introducción propusiste una duda, una pregunta actual, y en la discusión uno lee 2 páginas, 3 páginas de discusión y finalmente no encuentra la respuesta. Entonces hay muchos autores que parten con el primer párrafo diciendo, encontramos tal cosa en este estudio, y nos dieron la respuesta, y después uno lee la argumentación de esa respuesta. Entonces cuidado con eso, hay que dar una respuesta, pero fíjense en este otro, dice, explica tu respuesta, explica tu respuesta. No es simplemente decir, oye, hoy día va a hacer calor en Talca, sí, es la respuesta, sabemos eso. Y, pero la pregunta que sigue es, ya, ¿por qué crees que sí? ¿Te fijas? Diste una respuesta, pero tienes que explicar tu respuesta, y eso va a estar dado por tu capacidad de lectura, de análisis y la confianza que tienes en el estudio que hiciste en tus resultados. Ok, básicamente esto es más o menos lo mismo, era un taller diferente para estos dispositivos, así que no voy a entrar mucho en esto, pero la introducción hay que mantenerla corta, no como estoy hablando yo ahora, hay que describir brevemente la literatura, pero la más actualizada y específica, y no necesariamente los últimos 2 años, pueden partir 20 o 30 años atrás donde todo comenzó, digamos. Y eso es muy, muy importante. De hecho, algunos journals piden, por favor, vaya a la fuente original de esto. Y evitamos, la sugerencia también es evitar la cita de reviews donde se citó el trabajo original. No, vaya usted a leer el trabajo original y cuéntenos cuál es su impresión de ese trabajo en términos o en contexto de sus resultados. Algunas introducciones, aquí en el último punto, creo que dice, no haga un resumen en la introducción de sus propios findings, de su propio paper. Eso se usaba mucho en papers anteriores, hace 10, 20 años, pero hoy día no se usa mucho. Esto de terminar la introducción con, nosotros demostramos en este estudio que pasó tal cosa. Ya no se usa tanto, se usa más bien la pregunta como el mecanismo de impulso para el lector a ver cómo responde esta pregunta este individuo. Bueno, tips para éxito, eso yo creo que la discusión lo tienen, pero hay una lista de tips que pueden encontrar en cualquier lugar en realidad. Pero uno quisiera destacar es que asegurarse que las referencias a esa referencia o la discusión que hace esa referencia sea lo más aguda y precisa posible, porque es muy probable que uno de esos autores que tú citaste en tu trabajo, que son 40, 50, 60 autores, posiblemente a uno de ellos le llegue a revisar tu journal. Y vaya que sería desastroso encontrarse que yo publiqué un paper ayer y alguien me cita, lo cita todo al revés como yo lo publiqué. Entonces, a lo mejor se confundió el estudiante, el investigador, pero lo más probable es que no se haya confundido, porque tuvo un tiempo para leer y discutir. Y si no lo leyó y no lo discutió con precisión, se puede equivocar en cosas tan simples como esta, pero tan trágicas como esta. En papers puede que se solucione con un buen reviewer, pero en proyectos de investigación eso pasa por varias cabezas, digamos, con mucha experiencia, y la resolución normal es, bueno, aquí no hubo, ¿cómo se llama?, ser delicado en el análisis de la información. Y eso echa para atrás un proyecto. Y de hecho eso le puede pasar no solamente a la gente más joven, sino que a todos nosotros nos puede pasar diariamente, ¿no? Ok, como resumen de unos buenos consejos para publicar, está aquí mostrado hacer un estudio con, si quieres, solo para publicar un paper en vista de lo que quieres hacer, elegir buenos autores, contar una buena historia muy precisa, muy concisa y concreta, digamos, que no tenga ningún hilito colgando. Cada sección, método, introducción, es para eso, no repetir los métodos en resultados o no poner resultados dentro de los métodos. Eso la gente confunde la lectura, etc. Elegir un journal y pedir ayuda a colegas o compañeros de trabajo, que es algo que normalmente nos cuesta hacer, ¿no? Nos cuesta hacer. Cuando hay grupos más consolidados de trabajo, eso ocurre naturalmente, pero cuando uno está un poco solitario en el laboratorio, sobre todo un alumno nuevo que no sabe cómo funcionan bien las cosas, no conversa mucho con sus pares, o con sus postdoctors del laboratorio, por ejemplo, o con sus tutores. Y es ahí donde comienzan los problemas, el no comunicación. Y tener claro quiénes podrían eventualmente revisar tu trabajo. Entonces aquí, cuando un señor escribe en el periódico, escribe una carta o lo que sea, la escribe pensando en quién lo va a leer. Acá lo mismo, tenemos que pensar quiénes van a leer potencialmente tu trabajo. El proceso general es terminar tu trabajo de investigación, preparar el manuscrito, enviarlo, y en la revista, en las revistas, se asigna el proceso de revisión. Aquí pasa el editor senior con el editor en jefe, algunos regionales, editores regionales, y los revisores que toman este proceso para llegar a una decisión final. La decisión puede ser rejection inmediata, porque a lo mejor hubo problemas importantes que pocas veces que ocurre, ¿eh? O sea, uno diría, no, a mí siempre me rechazan los papers a la primera. No, eso no pasa siempre. Pasa mucho, pero no siempre. Pero cada vez menos, porque hoy día las editoriales quieren publicar por varios motivos, por aumentar la divulgación de la ciencia, pero también algunas de ellas por aumentar la posibilidad de publicar y cobrar $3.500. Entonces, ojo con eso, el nivel de rejection está disminuyendo en varios journals o se ha mantenido por muchos años. Después, normalmente lo que obtenemos es revisión. Haga usted una revisión de estos comentarios que hizo los revisores. Y ahí está muy bien, porque una vez que recibe eso, tiene la chance de poder corregir. Y en general, los buenos revisores hacen preguntas complejas muchas veces, o hacen preguntas simples, pero que son totalmente atingentes a tu trabajo. O sea, uno tiene que aceptar de esos revisores que esta pregunta que me hizo, a pesar de ser, uno dice, media maldita, es de verdad, ocurre. O sea, si tú estuvieras revisando, tú a lo mejor preguntarías lo mismo. Entonces cuesta aceptar, pero en ese proceso, cuando ya recibiste la posibilidad de revisar y reenviar, es que vas por buen camino en tu trabajo. Se hace un reenvío, se revisa nuevamente y puede finalmente terminar en aceptado. Aquí no hay una línea que diga, después de haber hecho una revisión, que pase a... no, sí está, perdón. La revisión puede pasar a aceptado inmediatamente, en muchos casos, y a un rejection, que muy pocas veces ocurre, ¿ya? Cuando uno contestó a los revisores no muy eficientemente, o simplemente a la rápida, o simplemente dijo, no, esto no lo voy a hacer porque no es el scope de mi trabajo. Bueno, ahí hay 3 personas más leyendo tu trabajo y a lo mejor encontraron que era parte del scope. A lo mejor uno no se hizo la pregunta, ¿será o no parte del scope de mi trabajo? Entonces, el rejection todavía ocurre, pero yo diría que cada vez menos también. Finalmente son aceptados, puede que eventualmente pase una segunda revisión, ¿ya? Tú contestas, el editor encargado se lo manda a los revisores para ver si está de acuerdo los revisores, y algunos de ellos pueden decir sí, otros pueden decir, ¿sabes qué? Bien, pero todavía le falta la pregunta 4. Entonces uno lo manda de nuevo a los autores y el autor toma esa pregunta 4 y decide si la contesta o no. Normalmente la contesta, ¿ya? Así que llegamos al éxito de la publicación. Es un proceso relativamente simple que toma 2, 3, hasta 4 meses, depende de la revista o más, pero es algo que hacemos todos los días, es nuestro trabajo, ¿no? Para un editor es muy importante saber a quiénes va a elegir para enviar tal tipo de trabajo. Entonces, lo que hace un editor es leer ese trabajo que le llegó. A lo mejor ustedes no saben, pero a uno le llega un paper y uno podría leer el abstract y un poquito de los métodos quizás y mandárselo a un revisor. Y eso es un error, eso es una cosa que nos dicen, y hay muchos que seguramente son editores, nos dicen los comités editoriales diciendo, no, pues usted lee el trabajo, vea si es atingente al journal y vea si quién va a ser el mejor revisor de eso según usted, pero no lea el abstract. Es como muchas veces cuando llega un estudiante novato diciendo, hice una revisión y tengo aquí 15 abstracts que leí. Claro, hice una revisión de los abstracts, pero no de los papers, ¿no? Así que cuidado con eso. Contestar en forma adecuada, creo que es muy importante, creo que en algún momento se dice también. Hay respuestas a revisores que algunos autores, quizás seguramente por descuido, como que pasan un poco el tono de su respuesta. Y uno como editor, cuando le llegan las respuestas de los autores, uno tiene que leer las respuestas y eventualmente editarlas para suavizar un poco las respuestas de algunos autores. Uno entiende que están molestos, les causó alguna insatisfacción los comentarios, pero no podemos tratarnos mal en el proceso editorial. Ustedes quizás han visto en las redes sociales algunos ejemplos de respuestas de editores o de revisores, perdón, a los autores, que han sido realmente motivo de discusión en muchos journals. De hecho, Journal of Physiology recibió un reclamo de uno de los autores hace poco, de uno de los autores, porque no se hizo el filtro de un comentario de un revisor a un trabajo en específico. Y fue agresivo, o sea, va y le dice el parágrafo número 1, no tiene sentido y yo no lo habría escrito así. Bueno, pero ¿por qué no eres un poco más amable? Y bueno, por favor corrija el parágrafo número 1 para que tenga un poco más de sentido con el contexto general. Pero hay frases difíciles, que la gente a lo mejor la escribe muy rápida, pero los autores hoy día están siendo más reactivos Yo creo que hace 20 años, 30 años atrás nos llegaba una cosa como esa y nos quedábamos un poco callados, ¿no? Pero hoy día, y lo que está muy bien, es el derecho a que te expliquen por qué tal o cual decisión. Ya termino. Aquí hay algunos ejemplos por qué pueden ser rechazados o las críticas más comunes de un manuscrito. Y aquí no los voy a leer todas, pero hay algunas como las típicas, ¿no? Que does not fall within the scope of the journal. Eso es típico y uno dice, pero ¿cómo? Y alega, pero podría ser. Eventualmente uno manda un set de resultados de, no sé, pues, del cardiomiocito, fisiología del calcio, al journal de, no sé, un extremo a fisiología educativa, no sé. Alguien va a decir, no, sorry, esto no corresponde con nosotros. No lo va a hacer, me imagino, pero poniéndolo como extremo. Pero hay cosas de ética que son muy importantes, como ustedes saben, especialmente con el trabajo de animales y hoy día con el trabajo en humanos. Así que esos son puntos críticos de rechazo y de crítica de los revisores. Si no hay comité de ética aprobado y abiertamente, muchos journals hoy están pidiendo el convenio de ética que se firmó con la institución para ver cuál es el procedimiento que se utiliza realmente, especialmente en el concepto de trato humanizado de animales. El proceso de revisión, ya veo que hay caras que están diciendo ya está un poco aburrido, así que lo voy a pasar rápido. Es responder a los revisores, vuelvo a poner aquí, es ser preciso y justo y convencido de lo que estás diciendo como respuesta, pero al mismo tiempo ser bastante diplomático en dar tu respuesta. ¿Ya? Y eso va a pasar solo si tú hiciste el estudio, si hiciste el análisis, si entendiste la pregunta que te hizo el revisor y si estás humildemente de acuerdo en contestar a una pregunta. También debo decir que uno recibe una crítica de un revisor y dice, este señor la verdad es que no leyó el paper. Uno podría decir eso también en una respuesta a los editores en forma diplomática. Y normalmente si un editor recibe eso, lo que hace uno es considerar ese feeling de ese autor y mandárselo a otro revisor. ¿Ya? No cancelar el primero, pero mandárselo a otro revisor para otra opinión. Así que es bueno hacerlo ver. No se queden callados cuando vean que crean que las críticas que le han hecho a ustedes las están contestando bien o simplemente el señor no leyó el manuscrito. Porque lamentablemente muchos de ellos no leen el manuscrito. Y es muy importante notar también y hacer notar aquí en esta sala y en todas partes que cuando a uno le llega un manuscrito y no tiene tiempo porque tiene reuniones o tiene el consejo de no sé dónde, se lo pasa ¿cómo se llama? Al postdoctor más viejo o más joven incluso para que le dé menos problemas, y el postdoctor lo lee y hace comentarios a veces muy buenos, muy precisos, pero un poco desequilibrado en términos de hasta dónde llego con mi crítica, ¿ya? Y después el autor, el big professor o lo que sea, manda ese informe como firmado por él. Y eso es algo que la política científica y los derechos de los investigadores están hoy día reclamando mucho. Entonces, cuando ustedes hagan un trabajo para un profesor, exíjale al profesor que identifique en su report al journal que fueron ustedes los que lo hicieron. Así de simple. Hay journals que piden explícitamente, ¿esto lo hizo usted o lo hizo su ayudante postdoctor professional? En general la respuesta lo hice yo, cada uno. Bueno, si hablamos ahora de publicar, quiero contarles por qué publicar en ciertos journals. Y aquí tengo un mensaje en mi rol de editor regional. La posibilidad de que ustedes publiquen en ciertos journals, voy a mostrar 2 o 3 editoriales y algunos beneficios de hacerlo. Pero antes de eso, si ustedes tienen alguna pregunta, alguna duda, podemos discutirla rápidamente. A pesar que ya estoy pasado. ¿No tienen alguna pregunta? ¿Sí? Por favor.
De la experiencia, ¿verdad? Y como mensaje para cuando uno manda un artículo a revisión, hay muchos errores del escritor y errores, digamos, pueden el editor manda a un revisor o a
juegue.
dirige un mensaje de decir, oye, tu proceso estuvo muy mal. A mí me detuviste 5, 6, 7, 8 meses, porque confié en tu revista, confié en tu grupo editorial, pero finalmente por un proceso humano y editor. ¿A dónde dirigimos?
Claro, ahí hay dos etapas. Una que es el editor, propiamente tal, por ejemplo, un editor regional o un editor de sección de la revista, que es el que maneja finalmente todos los comentarios de los revisores y manda al autor. Manda o sugiere al editor en jefe, este paper debe ser rechazado porque este es el informe de los revisores. Pero el editor en jefe asume que lo que hizo el editor asociado está revisado, digamos. Entonces el editor en jefe instruye al editor o a la revista que le comunica al autor que está rechazado el manuscrito en base a los comentarios hechos. Entonces cuando uno recibe una cosa como las que tú dices, le escribe al editor en jefe de la revista. Y aunque suene un poco para ustedes, para algunos de ustedes no, pero el editor en jefe no va a contestar nunca porque no están ni ahí con nosotros. Eso no es verdad. El editor en jefe de una revista sí está muy preocupado de lo que piensan los autores. ¿Ya? Entonces, cuando el editor no hizo su trabajo, ustedes consideran que no hizo su trabajo en forma adecuada y conversaron con el editor insiste, el paso siguiente, que es muy bien recibido por la revista, no es mal recibido. ¿Ya? Es muy probable que alguno piense, no, si le escribo al editor en jefe, estos dos son amigos, así que ya no me van a aprobar mi paper o no me van a dar otra opción. Mentira, eso no ocurre. El editor en jefe es súper tough, digamos, en decir, no, el proceso se tiene que reiniciar. O sí, sabes que leí todos los comentarios y tiene razón los revisores y lo siento, no se publica. Pero el camino es el editor siguiente en la escala, digamos. En general está el editor en jefe, los editores regionales, los editores ejecutivos y entonces los revisores. Pero hay que hacerlo, hay que decirle a la gente lo que queremos y lo que sentimos, amablemente. Porque si tú partes un texto diciendo, como dijiste, no, esta revisión está toda mal, bueno, eso es una cosa subjetiva, ¿no? Entonces hay que ser cordial, hay que ser cuidadoso, hay que ser profesional nomás, siempre. Pero el camino es ese, y el camino se puede revertir. ¿Mm? ¿Preguntas, comentarios de algún editor o de algún estudiante que tenga alguna situación? ¿Ninguno?
Un par de comentarios, sí.
¿Sí?
Lo que tú estás mostrando acá, la presentación del Journal of Physio, bueno, de fisiología, probablemente la revista más antigua, y lo tienes del 1878, han publicado muchos premios Nobel, pero es más conocido por los trabajos de Hodgkin y Huxley, que descubrieron los mecanismos de potenciación. Es el Journal de Hodgkin. El de Huey Bessel, que estudia visión, Huey Bessel. Es de Inglaterra, es difícil, así que les vamos a volar la palabra a la señora Kim Barr y a usted, que, como dice la canción, with little help from my friends. Es difícil, pero es muy riguroso, porque tiene esa tradición. Entonces, para nosotros desde acá, de un programa regional, es difícil, pero lo vamos a tratar, ya que se está presentando esto, recogiendo todas estas recomendaciones, lo vamos a intentar en algún momento dado. Sobre todo, muy importante lo que dices tú, ese trabajo que probablemente nosotros tampoco hemos hecho, es de discutir el manuscrito entre colegas, entre otro colega que lo revise. Eso sí, creo que es atingente, una obligación de hacerlo para que el manuscrito salga en las mejores condiciones posibles. Eso sí, hay que hacer trabajo especial. Paola.
No lo reconozco.
Hay tablas publicadas, de hecho, por algunas editoriales mismas. Hay una asociación que se llama Asociación Internacional de Publishers, ¿ya? Que la vas a encontrar en internet, y ellos tienen dos ramas. Una que es favorecer cómo encontramos el mecanismo para cobrarle más a los autores, y la otra que es cómo encontramos un mecanismo para favorecer publicaciones, ojalá gratis. Entonces hay dos peleas en esa asociación. Yo ingresé a esa asociación hace unos 4 años, 5 años, y la verdad es que pensé que era solo para beneficio de los autores, pero al final me di cuenta que ahí están divididos los grupos. Pero esa gente publica tablas, y las tablas son muy decidora en cuanto a números concretos que están en las propias editoriales de cada una de las revistas. Por ejemplo, en los últimos 2 años se han retirado de publicación más de 12.000 papers. 2-3 años. 12.000 papers, cuando hace 4 o 5 años era alrededor de 800, 500 papers. Entonces el grado de estrés ahora para cómo está funcionando tu publicación es mucho mayor, y eso ocurrió porque el grado de severidad en términos de revisión de tu trabajo ha sido mucho menor. ¿Y por qué es menor? Porque queremos ganar más. ¿Y por qué aumenta el número de rejections, no de rejections, sino que de sacarlos de una vez haber sido publicado? Es porque se ha dado cuenta la comunidad científica que los resultados no son válidos. 12.000 papers en 2 años, y aún más, si tú ves esa cita que está en literatura, de esos 12.000 papers hay 10.000, un 80% aproximadamente, que son de journals que hoy día publican lo que envíes, digamos, siempre y cuando pagues. Entonces ahí están los journals o las editoriales asociadas a ese tipo de publicaciones. Quizás ese es el camino para ir descubriendo qué son. ¿Okay? Bueno, publicar en el Journal of Physiology es para muchos muy bueno, por supuesto. El journal vino, la editora en jefe, Kim Barrett, vino el año pasado a Panam, en Puerto Varas, y conversamos con mucha gente. Y la verdad es que la propuesta del journal sigue siendo la misma de siempre, digamos. Tiene toda una historia, no cobra por publicar, es algo que muchos desconocían, me impresionó eso. No cobran por publicar, no hay restricciones en la longitud del artículo que se publica. Hay muchos journals que dicen no más allá de 6-8 páginas. Aquí no, puedes publicar lo que quieras, el número de figuras que quieras, siempre y cuando al tiro. Dame un segundo. Se hace mucha promoción. ¿Quieres que apure? Sí. Hace mucha promoción del trabajo que se está publicando, y especialmente vienen con la intención de ayudar principalmente a gente más joven. Así es que publicar en el journal puede ser complejo, como bien dice el doctor Durán, pero tienen la intención de facilitar además las posibilidades de publicación de la comunidad latinoamericana, y esa ha sido una de las razones por las que conversamos para poder incluir. Yo, cuando acepté este cargo o lo que sea, dije, bueno, pero hay un proceso para nuestra comunidad que tenemos que apoyar, y si no, ¿para qué tenemos un regional de latinoamericano? ¿Solamente para que aparezca en la revista? No. Es decir, queremos potenciar la investigación latinoamericana a través también de este journal. Y entonces, con datos en manos, ellos se dieron cuenta que, por ejemplo, el año pasado solamente hubo 30 publicaciones de Latinoamérica en el Journal of Physiology. En 2022 hubo 20. Y me dieron la tabla completa. En los últimos 15 años había cada año 20, 15, bajando hasta 10 los primeros años. Entonces, tener 20 papers de toda la comunidad latinoamericana, donde hay destacados fisiólogos, partiendo por Chile. Chile tiene una fisiología importante. Argentina, ni hablar, los brasileños. Entonces uno dice, ¿cómo esta gente no publica en un journal tan importante como este? ¿Por qué? Eventualmente porque no se les daba el acogimiento apropiado. Hoy día, Kim Barrett, que es la editora en jefe, tiene ese objetivo en los próximos 4 años de facilitar esta publicación. Había una pregunta ahí atrás y ya termino. Que llegue a golpear la puerta donde quiera hacer algo y hable con el tutor. Y el tutor, aquí la recomendación no solo para ti, sino que también para el tutor. Y el tutor tenga, ojalá, la capacidad de ver en ese individuo que llega así, casi en blanco, a un laboratorio, y reconocer en él la posibilidad que puede hacerlo bien. O, si es un buen tutor, la posibilidad que puede hacerlo mal. ¿Suena contradictorio? ¿Yo aceptaré a alguien que pueda hacerlo mal? En general no, porque uno busca avanzar rápidamente y dice, no, este señor no tiene, o esta señora no tiene un gran currículum como para entrar a mi laboratorio. De acuerdo a tus necesidades, pero en general un académico, como academia, va a tomar a un alumno en cero y lo va a, comillas, formar en el camino, le va a dar oportunidades. Una experiencia, un día yo recibí un estudiante que tenía un 4 como promedio en una universidad, bioquímico, y lo recibo como estudiante de tesis. Y mis colegas se acercaron y dijeron, pero ¿cómo tomas al peor estudiante de los estudiantes de bioquímica? Es el peor de todos, es el más bajo en la lista. Y, bueno, hoy día es neurofisiólogo trabajando en la Universidad de Chile como profesor asociado. Así es que no tiene sentido, simplemente el estudiante tiene que ir, tocar las puertas y ver. No tienes que llegar con un proyecto porque eres estudiante, estás partiendo, tienes que ver en qué le puedo ayudar, profesor. ¿Cómo podemos desarrollar un trabajo que me potencie? Y el profesor le puede decir, ya, un buen technician, por favor trabaja en esto. Ese no es tu mejor profesor. Pero el profesor, si te dice, bien, trabaja en esto, pero de aquí a 6 meses o de aquí a un año tienes que tener algo publicado con nosotros, si no, no tiene sentido que hagas todo este esfuerzo. Ese es el mejor camino. Buscar al mejor tutor. Así como buscamos el mejor journal para publicar, busquemos el mejor tutor también. Y uno está todos los días buscando tutor. Yo estoy todos los días buscando mi tutor. ¿Ya? Estaré viejo y todo, no tan viejo, pero estoy más viejo que ustedes. Y todos los días estoy buscando con quién conversar, con quién encontrar nuevas ideas, con quién explicarle lo que yo estoy pensando y que me la destruyan en 5 minutos. Y eso pasa siempre. Ok, yo creo que las ventajas de publicar en el Journal of Physiology son muchas y una de las cosas que uno no conoce es que son gratis, en primer lugar. Hay tiempos de demora, aquí dice 28 días en la primera revisión, que puede llegar hasta 3-4 meses, como dice aquí el profesor, pero va a depender de los problemas que surgen en el proceso editorial. En general es esto de 28 días. Pueden hacer preguntas, hay special issues, y hago propaganda de un issue que estoy editando, de hecho, que tiene la característica de tener un fast track en el proceso editorial. Entonces entra a un special issue como este u otros, y el editor encargado de eso impulsa ese grupo de papers lo más rápido posible y con la misma rigurosidad del journal, pero hace un fast track de tus papers. Así que es bueno hacerlo. Ya, como me estaban apurando, terminé con journal, pero por favor denme un segundo, si no me matan. Aquí voy a mostrar qué pasa con Benham Science. Benham Science es otra publisher que está también involucrado, y aquí ustedes eventualmente conocieron al señor Franz Letterstrom, bioquímico, que vino y estuvo en Panamá hablando sobre Benham Science. ¿Y qué está ofreciendo básicamente Benham a Chile? Ojo, a Chile, no a Latinoamérica en general. Y está ofreciendo acceso a 127 journals y 300 libros con licencia para universidades en Chile, incluida la Universidad de Talca, licencia que significa 0 costo por Open Access y que significa 0 costo de carga por proceso de artículos. Algo que encuentran todos los días en la internet. Cuando uno quiere publicar, dice Open Access 3.000, APS, no sé, 1.500 más, y suma 3.000, 4.000, 5.000 dólares. Entonces lo que hace Benham con el plan de potenciar Latinoamérica otra vez es ofrecernos a la universidad chilena, algunas universidades chilenas, incluido Talca, pongo, porque ayer conversé con Franz y me dijo, por favor, ofréceles la posibilidad de publicar gratis a la Universidad de Talca, toda la universidad, en cualquiera de estos journals, al menos por una marcha blanca de 2 años, y ver cómo funciona el número de publicaciones y después ver si llegamos a un acuerdo. Por otro lado, Benham está haciendo un agreement con una entidad que se llama Cincel, que se refiere básicamente a la biblioteca electrónica de información científica que maneja ANIT, y están a punto de firmar una renovación del contrato, que significa acceso a información libre por todas las universidades, que es otra historia, acceso a información libre. O sea, si yo hoy día quiero bajar un paper de un determinado journal, tengo que pagar también una fee en muchos de esos journals. Pero el consorcio Cincel está hoy día firmando o renovando el agreement con Benham para bajar papers en forma gratuita. Pero más allá, más importante que eso, es que de estas 127 revistas del Benham, que varían entre 2 puntos de impacto a 8 o 10 puntos de impacto, no tendría costo para la gente en Talca y en algunas universidades chilenas como la Chile, la Católica, etc., que se está firmando. Elsevier es otra editorial y también incluye el publicar a través de Elsevier para Chile un Open Access, ¿ya? O sea, ustedes pueden publicar en Open Access en Elsevier en muchas de estas revistas, y ahí están listadas las revistas que estarían con esa posibilidad sin tener que pagar, digamos. También el APC es cero, y se puede hacer un agreement específico con las universidades, con una, con dos, con tres universidades, como se quiera hacer. El número de autores, el número de publicaciones por autor o por universidad no está restringido. O sea, si esta universidad quiere publicar 10 papers al año está bien, si quiere publicar 100 o 1000, la revista no le va a poner problema, en cualquier área. Así es que si quieren conversarlo después con Elsevier o conmigo a través de Elsevier, podemos ver para estos journals que son bastante bien reconocidos, digamos, los BBA en general. Por ejemplo, Reviews on Cancer, que tiene 11 puntos, si les interesa tanto así el punto. Y así, etcétera. Está, por ejemplo, Molecular Aspects of Medicine, que tiene también 10.6 puntos, y otras revistas. Así que por favor no sientan miedo de publicar en revistas que parecen caras o que parecen imposibles. No todas son caras. Y hay estos dos convenios, Elsevier y Bentham Science, más el Journal of Physiology, como opciones para ustedes, a cero costo. Biochemistry and Biophysics es una Reports, es otra revista de Latinoamérica que está impulsada por Elsevier y que está hecha específicamente este issue está hecho específicamente para investigadores latinoamericanos residentes en Latinoamérica o aquellos residentes en el extranjero que sean primeros autores de journals. Así que utilicen las opciones. Estas son publicaciones, otra vez, gratis y totalmente indexadas. Bien. Si no hay preguntas, ¿no? Entonces les presento la otra parte. Lo siento, ¿eh? No es mi culpa, ni de ustedes. Ya, entonces ahora hablo de lo que uno hace para poder ver si puede publicar en esta revista. Hoy día es mucho más fácil porque la selección que hacemos nosotros, por ejemplo, en el laboratorio es buscar revistas en las que no tengamos que pagar. Primero, porque no tenemos dinero. Y segundo, porque particularmente yo creo que uno puede publicar en revistas buenas sin tener que consumir dinero de tus proyectos. Aquí me dicen que cambia inglés, ¿sí? ¿No? Votemos, po. ¿Inglés o español? Ok, en alemán. Okay, so we have 20, 25, 30 minutes to talk about this, and I'm going to show the endothelial dysfunction in a condition that we call gestational diabesity. So many people is asking normally, what is gestational diabesity? I think it was a mistake when you wrote the title in here, it's gestational diabetes. Then we said, yes, it's gestational diabesity, and that means that we have a woman that is obese and is pregnant and developed the gestational diabetes mellitus during pregnancy. So that is the definition of gestational diabetes. So I'm going to show what is happening with this baby growing here and the consequences for this pregnant period and the diseases of the mother or of the pregnancy and the consequences for the fetal physiology mainly. This is important because gestational diabetes is really affecting worldwide, and here we have some numbers. For example, in North Africa, 15% of the pregnancies are with gestational diabetes. In Chile and Latin America, it's around 15%, 16%, depending on the region. North American also, etc. Everywhere we have gestational diabetes as a big issue now. If we go back 20 years ago, 25 years ago, gestational diabetes was not a disease of pregnancy, it was just another condition. But then since then, it's a disease, and unfortunately still now the studies in gestational diabetes are not very much large in quantity and quality after 25 years. So it's important to know what is happening with this disease in Chile that I will show you is growing very fast also. This is a disease of the pregnancy. It's characterized by its recognition during pregnancy, and due to glucose intolerance or the incapacity of the mother to regulate the glycemia with diet, with exercise, or with some medicines. And gestational diabetes has perinatal risk, like we saw many times and still seen in the literature, microsomia of the individual that is growing, hyperglycemia of the fetus, and in some regions of the world still with fetal death, unfortunately, due to gestational diabetes, which is something that normally is not happening worldwide, but still we have it. And as you might know, we just had a macrosomic baby in Chile of 7 kilograms in the south of Chile, and now it's in Santiago. He or she is trying to survive, and the medics are working very hard with that. But 7 kilograms is macrosomia baby, very hard. And gestational diabetes is associated with around 20% of the pregnancies and macrosomia. So not all the pregnancies in gestational diabetes will result in macrosomic babies, but 20%, around 20%. The postnatal risks are many, and here we have risks for the women and risks for the offspring, and many diseases like kidney diseases or fatty liver diseases are associated with gestational diabetes. And in the offspring, you have obesity in very early in age, after birth, metabolic syndrome, and others. But one of these important modifications of the physiology of these patients, the woman and the baby, is what happened with the vascular system. You know, the vascular system is altered in gestational diabetes, and to study that, we started very early. I would say, well, I started my PhD, and many other groups are studying at the moment, and there's a large consortium worldwide studying the placenta as a main source of modifications and consequences for the baby that was born from gestational diabetes. And here we have microcirculation in the placenta to get endothelial cells, which is one of the targets of the gestational diabetes, or in general the diabetes is associated with endothelial dysfunction. So in the placenta, we can isolate microvascular endothelia from the microvasculature in the villus tree in there, or the macrocirculation, which are the umbilical cord vessels. And here we use the human umbilical vein endothelial cells, because these cells are seeing or sensing the blood with oxygen, with nutrients going from the placenta to the fetus. So it's carrying arterial blood in a vein, as unusual. So we will look at the endothelial dysfunction in gestational diabetes and other diseases. The first idea was to look at this pathway, arginine entry to the cells and arginine metabolism to nitric oxide mainly via the, for example, in this case, the endothelial nitric oxide synthase that I'm sure you know. So having this sequence of reactions or mechanisms, we are defining a proper endothelial function. That was the original description of or the meaning of the endothelial dysfunction. But we know now that any alteration in this project or in this link sequence of reactions will result in endothelial dysfunction. So not only a reduction in the synthesis of a NO, as the people was describing, but it could be also an increase in a NO. That is called dysfunction of the endothelium also. So don't keep the idea that endothelial dysfunction is a reduced production of NO. That's a very old definition. Endothelial dysfunction is an alteration of the function, any mechanism you're seeing in the endothelium. In this case, it could be up or down. So we want to see what happened with this mechanism in response to some agents, and I'm going to show you the results on insulin, because I was here last week, I think, giving a lecture to the students, so I mentioned I'm going to show the insulin effect in the endothelial function that we are describing. And insulin was shown to be a very good vasodilator in humans, as is shown here. You put insulin here, and after some minutes you have a vasodilation of the leg blood flow here, increasing the leg blood flow. But if you do the same experiments with LNMMA, which is an inhibitor, general inhibitor of nitric oxide synthase, there is not a vasodilation, but it's in fact a vasoconstriction. So insulin is doing something in the vessels to relax it and needs nitric oxide. If we look at the sequence of mechanisms described for insulin and nitric oxide, here is an example from Roberto Villalobos, a PhD student in the lab, now in University of Valparaiso, when he looked at the effect of insulin via these signaling mechanisms involving mainly protein kinase B or AKT to increase nitric oxide synthase at expenses of increasing the arginine transport in the cells via these proteins, resulting in increased nitric oxide and increased vasodilation. So the mechanism that insulin was shown by Baron and other studies early in the literature may include this sequence, including the insulin receptors 1 as indicated here, in the insulin receptor substrate 1 for insulin receptor A or B. So how insulin is ending in an increase in the vasodilation via nitric oxide, one of the mechanisms will be increasing the uptake of the substrate for nitric oxide synthase. And the substrate typically described is L-arginine, and L-arginine gets into the cell via these proteins, human cationic amino acid transporters. The isoform 1 is the more prominent in these cells. So we look at the effect of insulin on arginine entry in these cells. And what we saw here is this is the kinetics for arginine entry in cells from normal pregnancies in response to increasing concentrations of L-arginine in the extracellular space. So we found a maximal velocity of the transport and affinity or Km value for this Michaelis-Menten equation for the transport of arginine in cells from normal pregnancy. But now if we take cells from gestational diabetes, in general, this is a pool of samples in women with different characteristics, metabolic characteristics. In this case, the pool of samples include normal weight, pre-gestational normal weight of women, and pre-gestational obesity women in a mix. So we have these findings, which were published in 2017, based on results on 2015 and before. And what we saw is an increase in the Vmax in cells from gestational diabetes. So we have more uptake of arginine in these endothelial cells, and perhaps that could be, depending on the theory you will believe, in endothelial cells, we believe that the arginine entry in the cell is being directed to enos to use and then produce nitric oxide. That is the compartmentalization project or proposal from Professor Kloss in Germany many years ago already, which is now under discussion definitely because many groups have shown that not necessarily the synthesis of nitric oxide will depend on the entry of arginine, depending on the cells. Now, if we look at the effect of insulin in these cells, in cells from normal pregnancies, the yellow circles there, which is with the maximum of 0.4, Vmax 0.4 picomole microgram protein per minute of arginine. If we put insulin here, we have an increase of around 0.7. So in cells from normal pregnancies, insulin is increasing arginine transport, which is kind of logic, I would say, as expected. But then in gestational diabetes, insulin, instead of keeping the uptake increase, was reducing back to normal the values in gestational diabetes. So insulin is doing something different in cells from gestational diabetes mellitus compared with cells from normal pregnancies. That also sounds logic because it's a metabolic alteration in gestational diabetes. But what is surprising is that the effect of insulin was opposite to the normal cells or cells from normal pregnancies. If we look at the protein level of HCAT1, of course, we have something logic también for the experiments, and we look at the gestational diabetes, there is an increase in the protein abundance for HCAT1 in these cells, supporting the possibility that we have more activity due to more number of transporters at the plasma membrane, more than the change in the affinity, which didn't see in the kinetic experiments. And if we look at the effect now of insulin in these protein levels, we have a discrete but significant effect of insulin reducing the levels of HCAT1 in these cells. Same for the mRNA, but that's not very key now. So we have more arginine uptake and more protein being generated in these cells. If we look at the synthesis of nitric oxide in gestational diabetes, we have more synthesis of nitric oxide. And this effect would depend on was depending on the entry of arginine in our case, not in all the studies, as you can see in the literature. How the nitric oxide is being generated, one of the answers for that question was that in gestational diabetes, this bar here, the phosphorylation, the activator phosphorylation of phenos was increased from normal cells. And this activator, this is serine 1177 in the human isoform of, on human form of phenos. This increase in the phosphorylation means activity in the cells. The inactivator phosphorylation in threonine 495 is unaltered, so we have an increase in nitric oxide due perhaps to activation of phenos, and this effect was reversed by insulin, nicely there. So, however, I will say again, in cells from normal pregnancies, insulin was increasing the synthesis of nitric oxide, which is kind of logic, leading and explaining why insulin is perhaps vasodilating. But now adding that the vasodilation of insulin will require a arginine transport via HCAT1. But insulin is not acting only in one type of receptors. In terms of insulin receptors, we have defined from some years already that we have at least two isoforms of the receptors, receptor A and B, which are expressed in human fetal endothelium and the placenta in general. And they have different signaling cascades, or predominant signaling cascades, including the AKT and the MAP kinase P42-44. And these cascades are associated also with the arginine transport, as perhaps you remember from one of the previous slides. So we went to see where these receptors are expressed, and you can see that insulin receptor A is present predominantly in the placenta and in fetal tissues, and mainly at the end of the pregnancy when we use the cells after birth of the baby. And also it's involved in cancer and other conditions like brain function, etc. And insulin receptor B is more in the insulin target tissues like skeletal muscle, liver, and adipose tissue, for example. So the concept of having insulin acting in our body through one receptor perhaps is a kind of fault. We should consider now that insulin may act in A or B receptors, and this is the tendency now in the literature. If we look at the HUVEX, these cells and the mRNA for insulin receptor A and B, we don't have, there's no available still antibodies for the isoform A and B. But you look at the mRNA, in quantitative PCR, we found that the number of copies is increased in GDM for insulin receptor A compared with normal pregnancies, and insulin reverses that to values in normal. This was published early in 2011 by Francisco Estermeyer. He's now in Austria. And the insulin receptor B was unaltered in gestational diabetes and unaltered by insulin. So at the end of the day, we thought that insulin receptor B was not participating in response to insulin in the vasculature, but insulin receptor A, at least in the fetoplacental vasculature. If we look at uptake of L-arginine using 100 micromolar arginine, which is in vitro, which is similar to the circulating concentration of arginine in adults and in fetus also, in the fetal blood, we found that the uptake was increased, this band, the black bar here, but this increase was reversed by insulin, as I showed you, in gestational diabetes. But when the cells were not expressing or expressing very low levels of insulin receptor A, using an adenovirus against the insulin receptor A, the effect of insulin disappeared completely. This is insulin plus or in cells expressing very low levels of insulin receptor A. So we need insulin receptor A in these cells to reduce the elevated arginine uptake in this case, and transfer, as we will see later, in gestational diabetes. Okay. So, but if we do the same experiment now with insulin receptor B, knockdown cells, what we found is that the effect of insulin here, but the effect of insulin in cells lacking of insulin receptor B was still present exactly as nothing was done to the cells. So we need insulin receptor A, no insulin receptor B, for modulation of arginine transfer in the cells. We saw the same with the protein for HCAT1, so there is a correlation between the levels of generation of this new protein and perhaps the insertion in the plasma membrane, depending on the effect of insulin. These are effects of insulin for 8 hours, so we have time to relocate the transporters in the plasma membrane. Looking at the gene coding for this HCAT1, the SLC7A1 gene, we found in the promoter section that there is a here number 1 is like the control here, and here is the control for cells in gestational diabetes, which is around 1.5 of the luciferase activity compared with 1 in normal, so it's an increase in the activity of this section of the promoter. But then when we add insulin, the activity of the promoter comes back to around 1, which is control value. So also the expression of SLC7A1 gene in gestational diabetes in response to insulin is changing by insulin, modulated by insulin. So that may explain why the change in the levels of protein in these cells and mRNA. If we do the same experiment now with knockdown for receptor A, insulin has no effect, the red bars here. But if we do the same experiment with knockdown for insulin receptor B, insulin has a nice effect also. Where is it? Yeah. Where is it? Well, so in summary here, just to go even faster than I'm doing, the cells from gestational diabetes show more arginine uptake, more expression of the transporter, more synthesis of nitric oxide, depending on insulin in normal cells, and in gestational diabetes, these maximal effects or increased effect are moved back to normal levels in normal cells, cells from normal pregnancy. So using insulin receptor A and perhaps no insulin receptor B. Now, if we look at another component of this potential mechanism, we have here a nucleoside, endogenous nucleoside, adenosine. Adenosine is important because it's a good vasodilator also in almost all the vascular beds. And adenosine generates nitric oxide and other molecules acting on adenosine receptors in the plasma membrane. There are 4 subtypes of adenosine receptors. However, the biological action of adenosine is regulated in the fetoplacental endothelium by the capacity of the cells to take up adenosine into the intracellular space by nucleoside transporters. And this is apparently the only way that the cells from the fetal circulation have to regulate the extracellular adenosine levels. That's because they don't show any ectonucleosidase in the plasma membranes that will degrade adenosine to reduce the concentration. So if these transporters are not working properly, then the concentration at the extracellular space will change, and also at the intracellular space, because these are equilibrative nucleoside transporters. Their tendency is to equilibrate with the intracellular concentration. So we want to see what happened with these transporters in gestational diabetes, and what we found, this is normal, the uptake of adenosine, but in gestational diabetes there is a reduction of 50% of the transport. So there is lower uptake of adenosine in these cells, and remember, there is a higher uptake of arginine in these cells and nitric oxide. And if we have lower uptake, the question was whether this will result in an increased extracellular adenosine concentration in this model. We measure adenosine concentrations in the umbilical blood, including the whole blood with the veins and arteries, and we found that in gestational diabetes there is a concentration of adenosine in that blood which is double, or a little bit more, than in normal pregnancies. And this one was exactly the same when we look at only umbilical blood from the vein in these subjects. We found an increase of adenosine, but not in the arteries. So remember that the arteries are bringing the blood from the body of the fetus to the placenta, and from the placenta the vein going to the fetus with new and fresh blood purified in the placental barrier. So what we're seeing is that the umbilical vein is carrying a large amount of adenosine, but not the artery. So we are facing perhaps an abnormal function of the placenta in terms of regulating the concentration, the plasma concentration of adenosine. And that abnormal function of the placenta results in high levels of adenosine that are targeted or are sent to the fetus. And the fetus is receiving massive amount of adenosine in their blood, in their system. And after crossing all the body of the fetus, then you have a correction of adenosine. The fetus is trying to, and it seems to be done very efficiently by the fetus, correcting the elevated levels of adenosine that is receiving. But that means the need to metabolize adenosine by the fetus. But at the beginning, the fetus will be challenged with this high concentration of adenosine. And that what could happen is that adenosine, being a very nice vasodilator, adenosine increasing its concentration would result in vasodilation of the human umbilical vein also. And if you look, or if you have seen an umbilical vein from humans, the diameter is about 2-3 millimeters. So whatever we do as a relaxation on this tube of 6%, for example, 5%, will be very low in number, but in the amount of relaxation on a big tube will mean a massive amount of blood going to the fetus. So the fetus from gestational diabetes is seeing a lot of blood, perhaps explaining a kind of microsomia in this baby, because also carrying glucose and other molecules, and also adenosine. But not only, the blood coming from the placenta with information from the mother to the fetus is something that we know now, but it's also the blood from the fetus coming back to the placenta may affect the mother or maternal circulation also. And we are recognizing now for several years already, but with more evidence in the last years, that the molecules generated by the fetus will affect the mother, and perhaps some of these molecules or group of molecules will prompt the mother to be hypertensive in pregnancy, or perhaps gestational diabetes. We don't know yet. So it's not only a problem of the mother, but might be also a problem of the fetus affecting the mother. And if we look at here, we have the fetal capillary very much closer to the syncytial trophoblast of the placenta, the single layer of cells in the placenta that separate the maternal blood from the fetal blood. And if we look at the details in here, we have the syncytial trophoblast. This is the apical side or maternal side. And this is the basal side or fetal side, looking to the fetus. And here is the fetal capillary with the fetal blood. And what we think, and we were asking with Indira Chiarello, a postdoctor in the lab, some years ago, she asked whether this syncytial trophoblast is generating molecules going to the mother to alter the function of the mother, and whether this syncytial trophoblast is also generating molecules going to the fetal capillary and then the fetal blood to alter the function of the fetus. And one of the key questions was the extracellular vesicles generated by syncytial trophoblast. So we implemented the possibility of generating extracellular vesicle from syncytial trophoblast directed to the mother, and some of them directed to the fetal capillary, something that wasn't described in the literature. We cultured syncytial trophoblast in a double chamber, and we isolated in the apical side the solution to look up for the extracellular vesicles, particularly exosomes in there, and also at the basolateral side, which was the fetal side of these cells. And when we look at the CD73 expression protein in these exosomes, remember that CD73 is metabolizing AMP into adenosine, increasing even more the concentration of adenosine in the extracellular space in these endothelial cells. And we look at here that the exosomes from the basal side express the CD73. Basal side is the side going to the, or looking to the fetal circulation. However, the apical side, the exosomes from the apical side don't express apparently the CD73. So in this, look, just these results, we have the option of having exosomes in the mother circulation, maternal circulation, that are not synthesizing adenosine, but having exosomes in the fetal circulation that are synthesizing adenosine. And that's a very intriguing result, and we are trying to publish it, But when we look at the exosomes in an HPLC machine, we have generation of adenosine only when we use the basal exosomes with the substrate, but not when we use apical exosome, confirming a little bit the lack of CD73 in the maternal side. So the exosomes going to the mother from the cesiotrophoblast may be different in cargo that the exosomes from the same cell types going to the fetal circulation. What is making that these exosomes go to one way and not the other way is unknown. So that could increase adenosine concentration in umbilical vein. But also, we found that in gestational diabetes, and this is the picture here, yes, you can see a little bit. You can see that perhaps in the arrows there, the transporters for nucleoside transporters, for nucleoside I mean, are more concentrated in the intracellular space than in the plasma membrane like it was in normal cells. So like as a similar of glucose transporters, in gestational diabetes you have a sequestration of the transporters for nucleoside into intracellular spaces and reducing the number of transporters at the plasma membrane. This is in fact shown here where the number of transporters at the plasma membrane is reduced from 1.8 million of transporters per cell to almost half of the values. Also the expression of the SLC29A1 gene coding for this protein is also altered, showing that there is a decreased expression or activity of the promoter in gestational diabetes. Just going a little bit faster, if we look at a section in the promoter for gestational diabetes, in this gene we found that in this section of the promoter there is a binding site for this transcription factor, CHOP-CAP alpha complex, which is shown to be modulating the expression of this gene. And what we found in gestational diabetes was that in immunoprecipitation chip experiments, we found more binding to that specific sequence in there. And when this binding was done in cells that were lacking the expression or activity, in this case expression of nitric oxide synthase, this increased binding was reduced to normal and even a little bit more. So having more production of NO in these cells, you will have more binding of this complex to this sequence in the gene, and that will result in a reduction of the expression of the gene, and then a reduction of the expression of the gene will mean at the end lower number of transporters at the plasma membrane, lower entry of adenosine and accumulation of adenosine extracellularly. And I could be here talking another half an hour if you want, but I know you don't want. But I want to say that if we change the concentration of adenosine, we will modulate the large-unit nitric oxide system pathway in these cells. So having more adenosine extracellularly, perhaps because of this pathway, very well characterized by Marcelo Farias, now working at the Catholic University also, we will have increased concentration and increased large-unit nitric oxide synthesis in these cells. Right, I think I finished and we'll jump to one very important question here is that some studies, recent studies and reports from the literature here in Chile, we are seeing, as you know, that the obesity in women in childbearing age is increasing in Chile in this period. And if we look at the recent studies by Garmendia et al. in Santiago, de Chile, there is an increase from 2004 to 2020 around 20% gestational diabetes now in 1,000 women compared with 7% some years ago. So this is only 15, 16 years. And we increase it from 7 to 20% gestational diabetes in this group of women in Santiago, in a very localized place. And more important than that, I will say for our aims is that gestational diabetes from this group, around 40% of them were with obesity associated, obesity before pregnancy. So we told here in women that were showing pre-pregnancy obesity and developed gestational diabetes. And what Marcelo Cornejo did, Marcelo Cornejo is a PhD student here at the University of Talca in the program, is now currently in the Netherlands. He published in Acta Physiologica 2021 that the maternal pre-pregnancy BMI could configure different metabolic conditions or states of the mother. If she gets pregnant being normal weight, we'll have a classical, well-known GDM or GDM lean woman. And then if the mother was with pre-pregnancy obesity, he defines this as gestational diabesity. And when I say he, it's in fact a group. We have Marcela, Dr. Cavallini in Santiago that was involved, Dr. Pardo in Santiago también, also sorry. And then we have Paola Valero, that she's here as part of this contribution. So this was the kind of first definition of gestational diabesity in the literature, and from them we can collect several other papers referring to us, which is really good because we can see that the metabolic alterations in a woman with gestational diabesity are different from the metabolic alterations in a lean woman with gestational diabetes. Like here is some of the mechanisms we have been studying. Also, gestational diabesity was by Cavallini et al. Published in Molecular Aspects of Medicine, 2019, that show a disequilibrium or lack of equilibrium between pro-inflammatory and anti-inflammatory responses in the literature. And also, we found that adenosine, when it's increased in the extracellular space, could act on A2B receptor to be a protective factor for the inflammatory process associated with gestational diabesity. So, if we look at the something of the data you showed, just saw that yesterday at the poster presentation, here is results in basal PhD, basal intracellular pH in these cells separated by different groups with normal pre-pregnancy weight or with overweight or with obese women. So 3 different groups that we are trying to understand whether the alterations in the endothelial functions are same or different. And what we found here is that in gestational diabetes, there is alkalization intracellularly in women with normal weight, pre-pregnancy normal weight, and overweight and obesity. But obesity associated with gestational diabetes was in fact alkaline, but in a lower magnitude than for normal weight or overweight. So something is different in these patients, and if we look at the recovery rates, we saw exactly the same. There is a capacity of the cells to try to recover the pH, the intracellular pH, that is very similar in normal weight or overweight, but is reduced also in obesity. Looking at the components of this activity of the cells, we can see in some of the results by Gonzalo Fuentes, also a PhD student here at the program in Talca, and also he has just finished in the Netherlands for 2 years staying. He found that if we look at the patients with normal pregnancy, but that were obese before pregnancy, we found that the components of different mechanisms in the plasma membrane to regulate the intracellular pH was distributed between the transporters, sodio-proton exchangers, and other transporters like proton-potassium exchanger and ATPase potassium-proton exchange. So what he found is that in obesity, in gestational diabesity, there is an increase in the activity of this NH1 isoform, and also seen in normal weight pre-pregnancy, suggesting that if we look at the cells from pre-pregnancy normal weight, for normal pregnancy with GDM, we have a pattern on NH1, but also we have a pattern on other kind of transporters. So it's a question now open in the literature. If we now put the data all together, women with normal weight, overweight or obesity all together, and we do the experiments or the analysis of the experiments, we don't see the change in the NH1 activity. So it's very important to separate the cells from normal pregnancies or women with pre-pregnancy obesity. Then I finish with this. As you see, I have many other results I could show you, but didn't have time because the previous speaker used my time. And the summary is just this: fetal vascular dysfunction, endothelium, por favor, please, the message is just not just a depression of something, but it could be anything. The transporters are involved. These kind of transporters include the regulator of the intracellular pH. Now we have the nitric oxide. Another actor in this system is the hydrogen sulfide. Intracellular alkalization as a key mechanism in the cells. And of course, gestational diabesity to treat as a different kind of patients, not just a patient with gestational diabetes. No, it's a patient with gestational diabetes, as the literature is showing. Just to finish here is the people that did the work. Most of them, and large part of this work was done recently by Gonzalo, Paola, Marcelo, and then some other postdoctors in the lab doing other kind of things, and people associated with the lab in other countries, and also students. Some of the question was, how can a first-year student approach to a researcher? Here we have Antonia, which is already in the second year of medicine. She has been for 1 year with us. Fernanda, starting this year, or at the end of last year, also in medicine, and Andres, who did the nursery studies, just finished. So thank you very much, and the supporters, of course, for the meeting. Thank you.
Thank you, Dr. Sobrevilla, for two excellent presentations. I do apologize. I'm afraid we don't have at this moment time for questions, but you can ask questions to Dr. Sobrevilla during the coffee break. That's going to take place in the same place as yesterday. And thank you very much. We meet again here. We'll try to do a shorter coffee break. Okay? Que vamos a tratar de hacer un coffee break más cortito.
Our next speaker is someone who doesn't need any introduction. Is Dr. Andrew Quest. Very briefly, Dr. Quest got his PhD from the Swiss Federal Institute of Technology in Switzerland, and went on to be trained as a postdoctoral fellow at the University of Washington in Seattle, and then also at Duke University. He became also professor at that time in the University of Lausanne in Switzerland. At that time, he changed his focus of research to caveolin, a research focus that he has maintained till these days. Towards the end of the last, toward the end of the 1990s, he took a position as an associate professor at the Department of Cell and Molecular Biology, which is a branch of the Faculty of Medicine at the University of Chile. In 2005, he became a full professor at the same institution. During these years, he continued working on the role of caveolin as a tumor suppressor, and he discovered that caveolin was involved in many, in the regulation of many pathways that are involved in tumor progression. More recently, he has become very interested in the mechanisms through which caveolin regulates exosomes that promote metastasis. And even more recently, he also became interested in trying to understand how Helicobacter pylori promotes gastric cancer through the release of what he called outer membrane vesicles, which is very interesting topic because it takes a very local and very specific type of vesicles which might have some very interesting systemic effects in the context of a patient, for example, with gastric cancer. So without further ado, I would like to invite Dr. Quest to give us the talk entitled The Obesity Connection to Cancer. Thank you very much.
So how do I get started here? No, that's all mine. No, yeah. Okay, Nelson, thank you very much for the introduction. This is not working. Oh, I can't see it. Can anybody see the pointer? No, okay. So, ¿qué le pasó? No. Okay. Okay, so thanks again, Nelson, for the introduction. And as you can see, our research interests have changed over time, and many now converge on the importance of vesicles of different kinds in the genesis of disease, and so that's also something I'll be talking about today. So the discussion has been yesterday, you heard these three talks, focused on obesity-related changes, and my talk is sort of the last one in this series, and it's entitled The Obesity Connection to Cancer. And so Mariana already mentioned this yesterday, so I'm going to try not to fall off the podium here. So that there is a epidemiological connection between obesity and the risk of different types of cancer. So there are 13 listed here where this type of a connection has been established. I just want to highlight here breast cancer, where you can see as the BMI of people increases, you have an increased risk, the green line versus, for instance, the blue line, in developing breast cancer. And so, of course, the question that begs answering at this point is, how can we maybe come up with a mechanism to explain such, obviously this is a correlation, so an epidemiological correlation. There's no mechanism necessarily implicated. And of course the mechanism can be different for different types of cancer. Anyway, so one of the things we became interested in together with Mariana is this possibility that is illustrated in a review we published fairly recently, naming the possibility that, so these are adipocytes, and we already heard about the fact that they can, as they accumulate lipids, they sort of become progressively worse, and they start communicating with their environment, local and more distant environment, not necessarily in a very favorable way, and by liberating different kinds of molecules, as you can see here, they could potentially impact directly on tumor cells is one possibility. And there is another possibility that is illustrated here, because in the tumor environment we have what are called cancer-associated adipocytes, which are again quite different from the peripheral adipocytes. And so the possibility was revised here that these cancer-associated adipocytes may, in the tumor microenvironment, communicate either directly with tumor cells or indirectly via so-called tumor-associated macrophages with the tumor cells, and induce changes that would favor tumor development. So this is the basic outline concept of what I'll be talking about today. And I'll just summarize really briefly, so you know what the key message is. We spent a lot of time trying to implicate macrophages rather unsuccessfully, and actually at the end I'll show you a little bit about a direct connection we are uncovering now. So the question is then, how might these adipocytes, either here or actually in the tumor, if they're peripheral, then of course they need to communicate over a longer distance with the tumor, and this can involve molecules, cytokines, adipokines, and so on, or vesicles that are portrayed here, and somehow induces changes in the tumor, in the nascent tumor environment. So what kind of changes are we talking about here? We could alter the function of the immune system in the tumor microenvironment. That could be in a way that would be beneficial for tumor growth. Avoid inflammation. We could essentially induce angiogenesis, promote tumor invasion metastasis, tumor growth survival, and so on, matrix remodeling. So this all things that would then induce a more aggressive phenotype or favor the development of the tumor. So, as I said, I'm just going to focus on the connection to the immune system, and particularly the macrophage possibility. There might be a polarization of macrophages in a sense that would favor tumor development. And so, basically what we have here is a progenitor cell that can then become what is called an M0 macrophage, and then can either be polarized to an M1 phenotype or an M2 phenotype, using, for instance, different reagents. And so it turns out that this sort of black and white situation here, M1, M2, is not really very correct. It's actually a gradient. So you'll find cells that have maybe more M1 and less M2, and maybe more M2 and less M1 markers. And that doesn't necessarily help when you're trying to study this phenomenon. What is generally considered to be the case is that in the tumor microenvironment you have a prevalence of the M2-like phenotype. So what you want to see, if you stimulate these macrophages and you think you might be generating macrophages that will promote tumor development, you would like to see an M2-like phenotype. Okay, so how do these changes occur? I've already mentioned a few molecules there, but another possibility is that extracellular vesicles might be involved. So what are extracellular vesicles? These are these structures here, obviously like basically a type of liposome with a very complex content that can include microRNAs, proteins of all types, and obviously many different lipids. And so the idea is that these exosomes produced, for instance, by adipocytes might be able to determine what kind of phenotype you get here, macrophage phenotype. Now these EVs are known to obviously transfer molecules. I mean, this content then can be transferred to a recipient cell and can induce changes there. And there are many examples showing how these exposure to such vesicles can then induce signaling processes. So before we go on, let me just mention briefly exactly what exosomes are. Obviously, they're these vesicles that shed it from the surface of cells, and basically all cells, pretty much all cells, generate some kind of exosome-like vesicles. Now, the problem in working with exosomes is that many types of vesicles are produced by cells. So the big problem you always face is you've got to purify these vesicles, and hopefully you're going to get a preparation that will be at least enriched in exosomes, because you have in circulation, for instance, apoptotic bodies or oncosomes or ectosomes, and then last but not least down here, the small ones, exosomes. So these are about the size of a virus particle, so they're much smaller than the rest, but it's particularly, well, it's not easy to separate, for instance, exosomes from microvesicles. Microvesicles are released from the cell surface. Exosomes are derived from an intracellular compartment called the multivesicular body. So, problem here is exosome purification. Of course, there are many sort of procedures, protocols you can follow, like these ones here, low-speed spins to get rid of the larger particles, and these can then be followed by further steps, centrifugation steps, or you can buy columns and basically generate in that way exosome-enriched preparations. So how do you know you have an exosome-enriched preparation? You need to characterize those preparations. So one of the things you need to do is, for instance, this type of analysis, which will tell you how big they are. This is an example of, for instance, a nanotracking analysis. And you would expect something between 50, 100, 150. You can look at them by microscope, for instance, STEM, or you can just use a more sort of classic approach and do Western blot analysis. So you're looking at cell lysates here at exosomes, and then what you hope to see is that enriched in these preparations, you'll have markers of the sort of endosomal exosome biogenesis compartment, so CD63, TSG1, ALEX, and so on and so forth. And you want to exclude markers of the endoplasmic reticulum, like, for instance, calnexin. So those are the sort of criteria I will use throughout my talk. when I talk about exosomes. Okay, so here we are now, adipocytes. They produce exosomes, obviously, and they produce exosomes with microRNAs and many, many other proteins. And of course, one of them is this one here, which is my favorite protein, caveolin-1. And you see why it's my favorite protein in a minute. Anyway, caveolin is implicated in many things, also in adipocyte function. It's implicated in accumulation of cholesterol and lipid droplets, lipid synthesis, lipolysis, of course signal transduction, and as you will see in a minute, has a very complex role in cancer. Okay, so what's the role of caveolin in cancer? So this is this molecule. It's actually quite small, it's about 20 kilonewtons. It has this region that inserts into the membrane, and then it has a carboxy terminus and an amino terminus that look into the cytosol. The carboxy terminus has additional sites of anchoring to the membrane, so it's palmitoylated. So for signaling mechanisms downstream of caveolin, what we consider particularly important is this tyrosine residue here that can be phosphorylated. Caveolin has the possibility to dimerize and then oligomerize, and then it generates these types of structures which associate with lipid rafts, so enriching single lipids and cholesterol, and then generate structures like caveolin. So it has a structural role. But as I said, we're really interested in its role in signaling, in particular the role of tyrosine phosphorylation, tyrosine 14 phosphorylation. Okay, so now if we look at, this is the timeline for caveolin expression in tumor development in cancer. Most tissues at the onset express high levels of caveolin, if we're thinking about, for instance, an epithelial cell. And what we see in the transformation process is that caveolin will disappear. So it's associated, basically the loss of this protein is associated with initial tumor development. This loss here occurs via epigenetic silencing, so methylation of the promoter region. And what we then see later on, so in humans this might take 10, 20 years, when the tumors have grown to a fairly large size and become basically malignant, we see an increase in caveolin expression again. So this epigenetic silencing is reversed. And these cells now can be highly migratory, invasive, and metastatic. And so how can we explain this somewhat enigmatic situation? And this is a schematic we came up with a few years ago now. So what we found is that in early stages, caveolin is present in complexes with this protein called E-cadherin, which is obviously expressed in epithelium, normal epithelium, important for epithelial function. And there it collaborates in the sequestration of beta-catenin, which is an important cofactor which combine then to these transcription factors of the TCF-left family and induce the transcription of, also suppress transcription of some, but generally induce the transcription of many genes related to cancer. Here 3 examples: cyclin D1, cyclooxygenase 2, survivin. So regulates cell cycle, inflammation, cell death with survivin. And so of course, when we reduce these, the expression of these proteins, we have increased cell death and reduced cell proliferation. So this is how we explain the tumor suppressor effect. Now what happens, I said, over time then, after 20 or so years, then caveolin can reappear and has a completely different effect. And this is because when it reappears, here to the left, there is no E-cadherin. We've done experiments to show that you can take a very aggressive cancer cell with caveolin and re-express E-cadherin, and then it just cannot promote migration, invasion, and metastasis. So this loss of E-cadherin is a key step in gain of function of this protein, that in a way that will promote then migration, invasion, and metastasis via the activation of a small G protein called RAC1. What do we need to do this? Well, I already basically gave the story away in the sense that I already mentioned that this tyrosine phosphorylase, so this tyrosine 14 here is crucial to mediating or permitting caveolin to trigger these effects. And so we can generate mutations there, loss of function, or let's say constitutively active version. And if we do a migration assay or an invasion assay, you can see the wild type protein will promote migration and invasion in these B16F10 cells. If we introduce the null mutant, that will not happen, and the constitutively active mutant will favor migration invasion. So clearly this residue is important. Yeah, I thought I was missing something here. And so this is a more complex scheme now, showing how caveolin, when phosphorylated, recruits an adaptor protein that's called P85, which is actually the regulatory subunit of a PI3 kinase. And this is a gap for this small protein here called RAB5, so you sequester the gap and RAB5 becomes active. It then activates a GEF, a guanyl-nucleotide exchange factor, for the protein RAC1, which becomes active and promotes migration, invasion, and metastasis. And now why does this complex with E-cadherin prevent all of this? Because when caveolin is present in this complex with E-cadherin, a beta-catenin, it is predominantly present in a non-phosphorylated state, and that is due to the fact that a phosphatase is recruited to this complex called PTPN14 and dephosphorates the caveolin in this complex. So that's how we now can, in molecular terms, understand how the same protein can become a very potent tumor suppressor, and then, in a different situation, a very potent promoter of metastasis. Okay, so this is a timeline of our understanding of what caveolin does in cells. So we started off with cholesterol homeostasis, and then we've transited now through its role as a tumor suppressor and enhanced metastasis. And what has become a great interest in recent years, as I already pointed out, is the possibility that it will be important in extracellular vesicles like extracells. So all these mechanisms I discussed so far are what I call cell intrinsic signaling mechanisms dependent on caveolin. And so we'll turn predominantly to now what I call cell extrinsic mechanisms. Okay, so the question is, or was, these exosomes that are released, so the multivesicular body compartment, confuse here with the membrane, release these vesicles, and these vesicles are of endosomal origin, so they have contained certain markers, and the question also at some point was, do they contain caveolin? And if so, what does caveolin do? This is a somewhat busy slide. I'll try and lead you through it. So how do you go about understanding what a protein does in these vesicles? Well, you try and isolate vesicles that either contain or do not contain caveolin, because you knock the protein out in the cells that express the protein. And then you do a mass spec characterization of the vesicles. And so this is, in brief, what we did here. So we have wild type vesicles, short hairpin control vesicles, and vesicles from knockdown cells. And then you analyze the protein content, and then you look for possible differences in function that may relate to, for instance, then a more aggressive phenotype in a recipient cell, like adhesion and migration. And then so you get a short list of proteins, and you become particularly interested, for instance, in those proteins that are exclusively present in vesicles from cells with caveolin versus vesicles from cells without caveolin. And then your short list becomes even shorter. And then, of course, what you have to do is to confirm, this is all proteomics, so you have to confirm this with another approach, that is, for instance, by Western blot. And that's what we did here. So here you have vesicle from the MDA MB231 cells, wild type, short hairpin caveolin, so short hairpin control, short hairpin caveolin. And what you can see here is that, okay, these have caveolin, so caveolins include the vesicles, these ones do not. Other proteins are included essentially, so, well, this is actually the extract. I should have focused on this down here. So these are the vesicles. So caveolin is here in the vesicles, but not here in the cells that do not have the vesicles. And likewise, for instance, this protein CO61 or tenacin are present when caveolin is present, but not when caveolin is not present. This is for S100A9 is also partially the case. And so clearly what this tells us is that proteins are being included in these vesicles in a caveolin-dependent manner. And so then, obviously, the question is, when you see an effect in a recipient cell, is it really the caveolin, or is it these other proteins that are being included in a caveolin-dependent manner? So for that, this is another type of well, basically by America Campos, so I already mentioned this, and so we identified these proteins that are included in a more kind of caveolin-dependent manner, and then we have B16F10 cells, and we see other proteins that are included in a caveolin-dependent manner. So what this means is, depending on the origin of the cell, different proteins are included in a caveolin-dependent manner, and the vesicles then exert their effects in a manner that is dependent on the cell of origin. Okay, so now let's get back to our adipocytes. So here are our adipocytes, the vesicles, caveolin. So caveolin is abundantly expressed in adipocytes, as we've already seen all of this. And we want to know now, what is it doing in these vesicles? So for that, and the idea was, as I said, I started out, is that maybe these vesicles derived from adipocytes could somehow promote polarization of macrophages to an M2 phenotype. So of course you have to start out by isolating and characterizing these adipocytes. Obviously, these vesicles have caveolin, and then do proteomics and so on. So we're not quite, so I'll show you what we have in A and B. We haven't quite gotten to in detail to see yet. And then look at how they affect macrophages. So what you have to do here is induce differentiation of adipocytes using a protocol involving these reagents here. And you characterize the adipocytes at different time points. And so one way you can characterize differentiation is by using a simple marker here, the accumulation of lipid droplets that you can stain with oil red S. And here we can see the accumulation of these lipid droplets in the cell. So just using this microscopic assay, you can then say that you are really under these conditions, you are getting adipocyte differentiation. Then you can ask whether you are getting expression of different marker proteins in these adipocytes as you induce differentiation, and what happens to caveolin in this context. So just, for instance, PPAR gamma or adiponectin, they all go up. Caveolin simply seems to be maintained in a comparable level. This is the quantification, so of the different markers, and you can see how they increase, and you have a peak around 5 to 7 days for the different markers, and this is probably pretty much what you see also for caveolin. So there is a slight increase. It's not as pronounced as for some of the other markers, but there is definitely an increase, and so 5 to 7 days of differentiation is the time point we tend to work with, and then you go on and isolate, obviously, vesicles. So you know caveolin is in the cells. You isolate the vesicles. Here again, the NTA characterization. And you can see actually the best vesicles are the ones from pre-adipocytes, the best because of their size distribution. And then sort of the peak becomes less and less pronounced, and you have more and more peaks. So this would be larger vesicles here, which is not necessarily a desirable phenomena, but still you have a majority in this range around 150. And you can see in this analysis that the mode doesn't really change, and the mean also doesn't really change in terms of size. The size distribution, there is a slight change, so you have less of these vesicles of smaller size when the adipocytes, the pre-adipocytes are differentiated to adipocytes. So what happens now with the expression of these different marker proteins I already pointed out. So here we have the cells, here we have the vesicles, and you can see that these vesicles contain the markers characteristic of exosomes like ALEX, TSG 101 or CD81, but so this is in some lanes it's missing, but not necessarily the same amount of protein was loaded because it was actually quite difficult to isolate these vesicles. And these proteins like calnexin is not present. And well, we have caveolin in these vesicles. So it's in the cells, it's in the vesicles. Good. So now we go back to our question again. If we now incubate, generate these M0 macrophages or then M1 and look for M1, M2 differentiation, do we see an effect of these vesicles on these macrophages? So there are two ways you can do this. You can look at the appearance as marker specific for the different macrophage phases to polarization stages by flow cytometry. So you go through your protocol of, this would be peripheral blood macrophages or monocytes, so you enrich for these monocytes and then you induce differentiation to an M0-like phenotype, and then you incubate with other reagents to induce either M1 or M2 polarization or your EVs. And so what you expect or hope to see is a shift then in the profile you get off cytometry here. So these would be markers for, so this is a marker for M1 and this is a marker for M2 polarization. Now this sounds all nice and good, but using different types of macrophage preparations, this is just with peripheral blood macrophages again, we actually never were able to get really clear-cut differentiation to one or the other population. So the M0 population, the M1 population we supposedly got, and the M2 population, they all had M2 markers, which is of course not really good. And we did this with several markers and we got similar results. And in essence then, what we often saw in our experiments incubating them with vesicles, if instead of getting an increase in any one of these markers, if anything, we'd start getting a decrease. So this was obviously not very favorable for our hypothesis. So we tried another approach, which is this one here. Instead of looking at cell surface markers, you can look for the presence of specific molecules like TNF-alpha, interleukin-6, 1-beta, which would be characteristic of M1 macrophages, or interleukin-10, TGF-beta, which would be characteristic of M2 macrophages. And the same kind of protocol again. Now this worked well in terms of the controls, as you can see here. So if we look at sort of a differentiation protocol that leads to M1 macrophages, you have TNF-alpha, you have IL-6, and you have interleukin-1 beta, so that the positive control worked well. Obviously, with the same stimuli, what you do not want to see is an increase in M2, so that is, it's specific. So in terms of the cytokine markers we're seeing here, the polarization is specific for M1, or in this case, M2. So we're using interleukin-10 as a marker of M2 polarization. Again, here it's increase in M2, not in M1. So the controls worked really well. Unfortunately then, when we did the experiments with the adipocytes, we didn't see, at least in this case, any kind of M1-like polarization. Here we actually got something that suggests that maybe we might be getting some degree of M1 polarization. But in the experiments looking at the M2 marker, we didn't get any polarization to the, significant polarization to the M2 phenotype. If anything, we got suppression. So this is, of course, rather disheartening. Clearly meant that there was something wrong with our hypothesis. Either polarization was not occurring, or we were just not getting, or didn't have the experimental conditions that allowed us to see that. And in talking with people, so we did this with a group in Groningen that are experts on macrophage polarization, so Tos Damon, But talking more recently with immunologists, they actually said that the best way to go is just to look at macrophages in tumors. Okay, so that's what we'll do next and see whether maybe in tumors we do see polarization. But anyway, meanwhile, what we did, well actually Sophie did all of this, was to look at what might be going on with angiogenesis or something similar to. So would it be possible that these vesicles would just directly induce angiogenesis of tumor cells? And so for that, so of course you can say, well, why did we come up with that idea? And the reason is when we isolated exosomes from cancer cells, and we used this assay, which is this network formation assay using cells, so basically endothelial cells. This is a proxy for vascular genesis. And you can see that, for instance, with VEGF you get increased network formation. And with these wild-type vesicles, in this case they have, for instance, caveolin and tenacin, so these are derived from breast cancer cells. And if you quantify, there are programs you can use to quantify this, and then so you can see this is the positive control. With VEGF we have more network formation. Increasing the concentration of these vesicles, network formation goes up, to somewhat lesser extent with the control cells. And in this particular case we knocked down tenacin. Remember I said that tenacin was included in these vesicles in function of caveolin presence. So we knocked down tenacin. These vesicles still have caveolin, but they don't have tenacin, and so we showed here that network formation is dependent on the presence of tenacin in these vesicles. Okay. So we have an assay that is a proxy for then, in a tumor situation, angiogenesis. We tried now the adipocyte-derived vesicles, and actually we got very nice network formation, as you can see here. It's in a concentration-dependent fashion. And if we look at the quantification, the effect is comparable to what we get with our positive control. And it's, obviously this was a bit of a surprise, so it's the same in pre-adipocytes and in adipocytes. So these vesicles derived from adipocytes clearly have pro-vasculogenic effects in vitro, and we think this will also be the case in vivo. Actually, people say that if you get an effect in this assay in vitro, you'll definitely see something in vivo. But we haven't done the experiment. Okay, so we have now this situation where the adipose tissue somehow promotes angiogenesis or changes the behavior of endothelial cells in a way that would favor angiogenesis, without necessarily the need of macrophages, because this is an in vitro assay. And so the question now, and I'll come to a close here soon, how could this be happening? We published a few years ago this paper in Molecular Cancer, where we showed that a protein survivin, which I mentioned before, is a target for beta-catenin and TCF-left, is actually controlled by the presence of survivin in cells. So the presence of beta-catenin depends on this destruction complex here, which phosphorylation by GSK3 beta, and then destruction by this proteasome-mediated destruction, essentially. And so if you prevent this phosphorylation, then beta-catenin can go to the nucleus and promote here transcription of these genes. And what we found out is that actually we can sort of short-circuit this and just simply overexpress survivin in cells. And what we saw was survivin, by a mechanism we were not able to characterize in detail, could activate PI3 kinase AKT and essentially promote a mechanism that inhibited GSK3 beta dependent phosphorylation and therefore degradation of beta-catenin. So in a nutshell, augmented the presence of beta-catenin in the cytosol and the possibility of it going to the nucleus, promoting expression of these genes, and one of them was VEGF. So what we showed is that VEGF is then expressed in a survivin-dependent manner and can promote angiogenesis, and that we did in vitro and in vivo assays. Okay, so do do we have now surviving in these adipocytes? And to just keep it short, as you can see here, pre-adipocytes and adipocytes, they have surviving. This is the quantification. They also have caveolin, of course. And then if we isolate the EVs, so the question was, well, do we have now surviving in the EVs? And the answer is yes. So again, this scheme of differentiation here, pre-adipocytes, 5 days, 7 days, 9 days differentiation. Isolate the vesicles and look for these markers, amongst other things. And you can see that they have the markers characteristic of the exosomal compartment and also surviving. And calmexin is excluded. Okay, so we have vesicles with surviving. And so how do we now think that this might impact on angiogenesis or vasculogenesis, angiogenesis? So what we found in the literature is that leptin is also produced by cells involved in obesity, stimulates this receptor here, and this receptor activates PI3 kinase AKT and promotes beta-catenin TCF-left dependent transcription of genes like, for instance, VEGF. So what we think is happening is when these vesicles now fuse with endothelial cells, so these vesicles now containing survivin, they trigger this pathway, and in doing so promote VEGF liberation by the same endothelial cells, and thereby sort of in an autocrine-paracrine fashion promote vasculogenesis, angiogenesis. But this is still something we will need to show. This is our sort of current working hypothesis. So I'll just conclude there. And basically adipocytes produce EVs and exosomes with caveolin, also with survivin. We did not see significant M2 polarization, at least in human macrophages, and also done experiment with mouse macrophages, and it's the same. Adipocyte EVs promote this vascular network formation by endothelial cells in vitro, which is a proxy then for angiogenesis in vivo. And survivin is known to promote beta-catenin TCF-left dependent transcription of VEGF and liberation by cancer cells and angiogenesis. And so we think this similar mechanism will also be in play and explain how these vesicles can promote vasculogenesis and then in endothelial cells, or of endothelial cells. Okay, with that, I'd just like to finish here. So this is our group a few years ago, rather large, decreased in size since, but still in collaborating with many of them. And so I just mentioned the people that are involved in the studies that I talked about. So Vicente Torres, Lorena Lobos, Rino Ortiz, they all have their own labs. Natalia Diaz, America Campos, Jorge Diaz Herrera de Burgos, they're now doing postdocs, but we're still in other labs, in other countries, and we're still in touch too, working on different projects. And particularly highlight Sofia Sambuesa, who has made a heroic effect of trying to understand what these vesicles might be doing to macrophages, unfortunately rather unsuccessfully. Manuel Varas was important for the experiments in knockdown cell. Mariana Cifuentes, co-director of the thesis, of Sophie's thesis. Our collaborators abroad, Carlos Salavon, Jay Heineke, Bart Frey, different experiments using mass spec to characterize either vesicles or complexes, for instance, caveolin with ecadherin. Our financial support, so FONDAP funding for many years now, FONDACIT, ANIO, international grants. So, thanks for your attention. Be happy to take questions.
Thank you very much, Dr. Quest. So we have some time for questions.
La respuesta, la producción de moléculas por parte de los adipocitos es muy distinta cuando el adipocito está inflamado a cuando no lo está. También algo de eso nos contaba Mariana ayer. Entonces, la pregunta, ¿podrían esto que tú nos has mostrado, esta señalización, cambiar, por ejemplo, porque ustedes han probado solamente adipocitos wild type, ¿verdad? Sin estimularles, por ejemplo, con TNF o algo así. ¿Podría esto cambiar si estos adipocitos están inflamados?
So we did experiments, I believe, with TNF to sort of as a political control. So you're saying add TNF and then add the vesicles? That would be the experiment? That we didn't do. Okay. But yeah, it's a possibility.
Thank you, Dr. Quest. It is possible that this phenotypic change in the endothelial cells also maybe is not only in angiogenesis, but also in endothelial dysfunction, more adhesion molecules, exposition in the membrane, so making the next with the cardiovascular disease, maybe?
Maybe what?
A connection between pro-inflammatory adipocytes and not only angiogenesis, but also endothelial dysfunction and cardiovascular disease. Next, maybe, because you show a pro-angiogenetic effect,
Right.
but maybe also not only pro-angiogenetic effect, but also endothelial dysfunction, maybe.
Oh, so that might affect other organs, like,
Okay.
yeah. So that's obviously a possibility, and it's something we could collaborate with anyone working on cardiomyocytes or cardiovasculature. I'll be happy to.
Thank you very much for the presentation. I was thinking a little bit more in the clinic, since these EVs that comes from cancer cells, as I understood, can promote angiogenesis. Is it possible to use other EVs that can somehow nullify the effect of these EVs from the cancer cells? Is it theoretically possible, but I don't know if it's applicable?
Well, that's a very interesting question, and that's also something we've been thinking about. So we have some experiments, obviously not with adipocytes, but with vesicles derived from cancer cells, where we knock down different components, like, as I showed you, caveolin, or then tenacin, and so on. And particularly when we knock down caveolin, and we test those vesicles in in vivo, in an endometrial carcinomatosis assay, we see actually effects that would suggest that those vesicles are almost like tumor suppressive vesicles. So clearly, the inclusion of in a caveolin-dependent manner of specific factors is what will really drive then in these assays, in vivo assays, will drive the tumor formation process. And if these molecules are not there, the vesicles, I mean, they don't do anything. And they actually, it looks like they could actually suppress tumor growth. So this is something I'm kind of thinking of a project there, how we could potentiate that effect and maybe then actually generate vesicles that could serve to impede cancer growth and metastasis.
Thank you very much, Dr. Quest. So we give him a big applause again. And our next speaker is Dr. Victor Cortez. Victor Cortez is a physician and also a PhD doctor of medicine from the Catholic University in Santiago. Between 2006 and 2011, Dr. Cortez did a postdoctoral training at the Department of Molecular Genetics of the University of Texas in Southwestern Medical Center in the United States. Since 2016, Dr. Cortez has been associate professor at the Department of Nutrition, Diabetes and Metabolism of the Catholic University in Santiago. His line of research is focused on the biology of adipose tissue with emphasis on lipodystrophy and obesity and its relationship with lipid metabolism, insulin resistance, and fatty liver disease. So without further ado, I would like to invite Dr. Cortez to this podium to give his talk today, which is entitled Neuroendocrine Regulation of Food Intake and Energy Expenditure, an Integrated View of Obesity and Lipodystrophy.
Bueno, perdón por la tardanza, pero falló la tecnología. El pendrive no funciona. ¿Tiene un pendrive? Okay. Thank you for your patience. Thank you for the organizers for inviting me to this nice meeting, summer school. I changed the name of my presentation because the name that Nelson read was the title of our ENERO grant. So, and it's not my expertise. The neuroendocrine regulation of appetite. I know something, but I'm more expert on this subject, lipodystrophy and obesity. So today I'm gonna show you especially this condition called lipodystrophy because it's a rare disease, but I think it can highlight very important aspects of adipose function and pathology that can complement our understanding of obesity. So lipodystrophy is a heterogeneous group of adipose tissue diseases. In general terms, it consists of a severe decrease in the whole body adiposity. So the first 3 patients here, all these patients are Chileans. All of them have been characterized at the clinical, metabolical, and genetic aspects in our laboratory. So as you can see, the 3 first have a general leanness. These persons basically lack subcutaneous and internal adipose tissue. The last one corresponds to a different form of lipodystrophy. This is called partial lipodystrophy because it is preserved the adiposity in the trunk, but it is decreased in the face and the limbs. So I'm going to show you first, the first part of this talk, some clinical cases in order to illustrate you what this disease consists, what is it. This first patient is a woman. The first time I met her, she was 29 years old. Important points in her clinical history it's that she was born of consanguineous parents, first degree cousins. She was small at birth. The first manifestation of metabolic disease was a very early age, at 3-month-old age. Because this baby didn't rise properly, so the physicians took some blood samples and found that plasma was extremely hyperlipemic, meaning very high levels of triglycerides in plasma, in fasting plasma. At 11 years old, she became overly diabetic, severely diabetic. When I examined her, it was obvious that she was very lean. She has an intense subcutaneous and generalized lipodystrophy, but also she has these findings called xanthomas in the tendons and also in the skin, suggesting a severe hypercholesterolemia and severe hypertriglyceridemia too. Also was very notorious this intense acanthosis nigricans. It's not very well seen in this picture, but it consists in a darkness of the skin, in the neck, and the axilla, and in the inguinal folds. She also has acromegaloid features, meaning enlarged lower maxillar, enlarged hands and feet. She was lean, body mass index was very decreased, and interestingly, from the medical point of view, she has no hepatomegaly. You will see why is that important. To illustrate you what we're talking about the metabolic and biochemical abnormalities in this condition, you can see in these patients different values of glucose, glycated hemoglobin, triglyceride, and plasma cholesterol. These are the normal cut points. At 13 years old, you can see she has a severe hypertriglyceridemia and, more impressively, a severe hypercholesterolemia. These very extremely high levels of cholesterol are commonly seen in another sort of metabolic conditions, like familial hypercholesterolemia, because of specific abnormalities in cholesterol metabolism. But wasn't the case in this patient. She only has lipodystrophy. The diabetes was, as you can see here, very difficultly controlled. She was using, the first time we met, a whole set of drugs to control her metabolic abnormalities, and they weren't enough to normalize these parameters. From the point of view of eating behavior, congenital generalized lipodystrophy, which is the condition that this patient has, is characterized by severe hyperphagia. These patients report that they're hungry the whole day, and they never feel, satisfied, and they can eat anything because they're always hungry. It's from the quality of life. Obviously, it's a problem, but it's also a problem from the clinical perspective, because in the face of severe and uncontrolled diabetes and hypertriglyceridemia, you need to introduce modifications in the diet, but they fail to do that because this severe hyperphagia. This is a way to represent this hyperphagia. This is the average of normal women, 50 women, that we studied in our laboratory to sort of quantify eating behavior. All the parameters upper in this line are promoting eating, and in the bottom part of the figure are those aspects of the eating behavior that are suppressing, or decreasing eating behavior. So this is the regular and normal way to visualize this, and these patients, as you can see, have obviously biased eating behavior towards the pro-intake behavior. From the genetic point of view, we studied this patient, but also her sister, that was this patient reported was very similar in her overall leanness. And we found that both of them have 2 point mutations in the gene called IPA2. From the metabolic point of view, you can see here, it was interesting for us to know that even though these patients have this very same genetic abnormality, the metabolic phenotype was different. The patient 1, which is the one I already explained to you, it has a much more severe metabolic and hormonal abnormalities, including non-detected adiponectin and undetectable also, almost undetectable leptin, very little levels of circulating leptin, which is consistent with the severe decrease of the adipose tissue mass. But for whatever reason that we already ignore, her sister has lower levels of metabolic abnormalities. This is a different patient. We got here case 3. She was a much younger patient when I met her. She wasn't born of consanguineous parent, but in her family history was notorious the prevalence of several autoimmune diseases, including arthritis, lupus, and Sjogren. She was normal at birth, but at 4 years old, she started with manifestations of autoimmunity. First, she developed hemolytic anemia, and then thyroiditis, hepatitis, requiring immunosuppressive therapy. The mother reported that this patient, around the puberty time, started to decrease body weight. But curiously for her, she was manifesting a hyperphagia too. So it was very kind of paradoxical for the parents, because this girl started to eat a lot, but didn't rise. And on the contrary, she was getting leaner and leaner. At 13 years old, she manifested overt insulin resistance, requiring the addition of metformin, which is a drug used to increase the insulin sensitivity. And it's very common in the medical practice. At the physical exploration, she was obviously very lean, as the former patients have already showed, and also manifested intense acanthosis nigricans, meaning increased levels of circulating insulin. Different from the previous patients, she has no xanthoma in her skin. She was studied at UT Southwestern, and they didn't find any mutation in the known genes related to generalized lipodystrophy. Therefore, she has a acquired form of generalized lipodystrophy, which is a different syndrome, but similar metabolic and clinical manifestations. So going back to our first patients, I mentioned that these two ladies have mutations in the gene called IPAC2, which is a known cause of congenital generalized lipodystrophy. So these patients are born with this condition. It requires to be present these mutations in homozygous form. The heterozygous people have no lipodystrophy, and but they can transmit for sure the disease. IPAC2 is an enzyme involved in glycerolipids and glycerophospholipid synthesis. This enzyme, this family of enzymes, is catalyzing the conversion of lysophosphatidic acid into phosphatidic acid. From the IPACs, there are at least 11 isoforms known, each of them codified by different genes in different chromosomes. All these 11 isoforms of IPAC have diverse enzymatic activity, meaning they have differential preference for the species of lysophosphatidic acid and also the fatty acid CoAs that are required to form phosphatic acid. Also interesting, these isoforms have a diverse and selective tissue expression pattern. Probably this is important because only mutations in the isoform 2 are related to lipodystrophy. And concordant with this idea, IPAC2 is the main IPAC isoform in the adipose tissue, but also in the liver. So you can see this enzyme is very critical for the formation of very important structural and signaling lipids. To study this condition, during my postdoc in UT Southwestern, I developed this knockout mice, which we found was able to recapitulate every single aspect of the human lipodystrophy. These are the wild-type mice. These are knockout mice. You can see they lack subcutaneous, interscapular, and visceral adipose tissue, including mesenteric adipose tissue. These animals have hyperglycemia, hyperinsulinemia, hypertriglyceridemia, and decreased leptin levels, as the patients that I already showed. These mice are also hyperphagic and hypermetabolic. You know, mice are nocturnal animals, so you have to study them, their eating behavior and their metabolism by separate during the day and the night. We did this with metabolic cages that, by the way, we will be able very soon in the Catholic University to acquire this technology thanks to a FundAICUB grant that we have already in our hands. So these hyper-2 mice are hyperphagic both in the day when this animal normally don't eat or they eat a little, a lot less, but especially in the night. They also, for sure, take a lot of water because they are hyperphagic and because they are severely diabetic. These animals are really diabetic, so you can see their cages, the substrates, the soil of the cages all the time are wet because they are urinating a lot. And also have increased levels of oxygen consumption, indicating hypermetabolic and increased metabolic rate. Interestingly, the addition of leptin in the form of recombinant leptin in humans have been shown to be very important or very potent to decrease almost every single metabolic abnormality in lipodystrophy. These are the data of the first patient I present today, the case 1. As I'll show you, this patient has a severe diabetes and hyperlipidemia, but after the introduction of metroleptin via subcutaneous injections once a day, you can see basically diabetes disappear, or at least became very well controlled. Also this hypertriglyceridemia and hypercholesterolemia, doing anything else than injecting leptin. Okay. This animal model happens essentially the same thing. This is an experiment with the knockout, IPAC2 knockout mice. These are the wild-type mice, these are the knockout mice. The last group of animals receive leptin infusions by a continuous infusion using mini pumps, osmotic pumps, during 2 weeks. And you can see how this single intervention was able to restore insulin, glucose levels in plasma, but also triglycerides in the liver are decreased, and cholesterol in the liver. At the behavioral standpoint, metroleptin in our patient was able to normalize the eating behavior. Okay, it suppressed totally hyperphagia. If you remember, what this was the profile of eating behavior of our patients before metroleptin, but after metroleptin, just 1 month after metroleptin, this pattern isn't significantly different from the normal eating behavior. Same thing happens in the animals. These are 2 cohorts of knockout mice before and after leptin. These blue ones are the IPAC2 knockout mouse infused with PBS, which is the vehicle of leptin, and these are the ones infused with leptin. And you can see the food intake is significantly decreased after 2 weeks. Interestingly, though, these lipodystrophic mice have increased levels of corticosterone, which is the main glucocorticoid in this species. In humans, it's cortisol. In the mice and rats, it's corticosterone. You can see these knockout mice have several times, like almost 12 times increased corticosterone, and leptin, even though it doesn't normalize corticosterone, it significantly decreases. Same thing was found in another model of lipodystrophy. This is a different animal model of lipodystrophy, but it was shown that corticosterone is increased. Apparently, it's the leptin deficiency which is driving this excess of circulating glucocorticoids in animals, but probably also in human beings. We are studying that in our neuroendocrine grant. But in this animal, which are called OB because they lack leptin and they are severely obese, they have increased levels of corticosterone. The same thing is in this other mouse model called DBDB. These animals are also morbidly obese because they lack leptin receptor and they have elevated corticosterone. Interestingly, the surgical ablation of adrenal glands suppresses or decreases circulating glucose in the OB mice, but also allows to control the morbid obesity, suggesting that corticosterone or the glucocorticoids in more general aspect are implicated also in the metabolic abnormalities of leptin deficient mice, unlikely leptin deficient humans, like the lipodystrophic people. So in conclusion of this first part, lipodystrophy causes hyperphagia and insulin resistance and all these secondary conditions like diabetes, hypertriglyceridemia, and fatty liver. Leptin replacement is able to reverse metabolic abnormalities in lipodystrophic patients, but also in our animal models. And leptin deficiency associates with increased glucocorticoids, and we believe this is an important link in the actual and current model of eating behavior regulation and also metabolic regulation. In fact, the surgical suppression of the adrenal glands decrease hyperglycemia and obesity in the OB mice. I'm going to talk about a little bit of fatty liver. As I already said, lipodystrophy is associated with steatosis. Actually, it's associated with steatohepatitis. These animals and people develop inflammation of the liver in association with increased triglyceride content in the liver, as you can see here. I'm not going to show all the data that allow us to conclude this, but we found that the fatty liver in this animal models is driven basically because de novo lipogenesis. De novo lipogenesis is the formation of fatty acids and triglycerides starting from non-lipid precursors, basically carbons derived from glucose or fructose. So every single enzyme in this pathway is expressed here in red, and we measured these transcripts in the liver of IPAC2 mice and found that every single one of them is increased, including pyruvate kinase, which is interesting because the reason I'm going to explain immediately. Insulin is able to promote fatty acid formation in the liver via this transcriptional factor. This transcription factor called SRBP1C is present in the precursor form in the membrane of the endoplasmic reticulum. And after insulin activation for partially known mechanisms, this transcription factor is processed, proteolytically processed, and translocates into the nuclei to promote the transcription of all the enzymes that I already showed you. And that increased the lipogenesis. Interestingly, this SRBP1C, N means nuclear and P is precursor. This mature transcriptionally active form of this transcription factor is increased in OB mice and is in this other lipodystrophy model. This is a different one. It's not the one where we are talking now. As you can see, this is severely, notoriously increased. However, in the IPAC2 mice, we found that SRBP1C, but also other isoforms of this transcription factor are not increased at the mRNA level or protein level, indicating that there is something different in this animal. regarding or in comparison with other models of fatty liver and insulin resistance. But to study this point more directly, we generated a double knockout mice by crossing the IPAC2 with SRBP1C deficient mice. But essentially we found that this, the lacking of this transcription factor didn't modify the triglyceride content in the liver of these animals, or the plasma triglycerides, or fatty acid liver, free fatty acids, glucose, or insulin in the plasma. So confirming that the fatty liver in the IPAC2 mice is not mediated by this pathway, the insulin SRBP1C pathway. There is another transcriptional regulator of de novo lipogenesis. It's called ChREBP, carbohydrate responsive element binding protein. And this system is directly regulated by glucose, no insulin. Yeah? Glucose is a direct activator of this system. It doesn't bind to ChREBP, so there are several steps in between, but it is the main driver. So we did the same experiment, but this time we crossed the IPAC2 with the ChREBP deficient mice, and we found, in this case, that ChREBP deletion resulted in a significant decrease in the circulating glucose, insulin, although no modifications in free fatty acids in the plasma. More interestingly, from the hepatic point of view, the deletion of ChREBP decreases or normalizes the exaggerated levels of all these transcripts involved in the novel lipogenesis. And also, unexpectedly, we found that fatty liver was totally normalized with ChREBP, meaning that this is the pathway that is driving the novel lipogenesis in the IPAC2 mice. So in conclusion of this part, IPAC2 mice have fatty liver, and this is caused by exaggerated de novo lipogenesis. This seems to be also true for human beings. Normally the liver, the total pool of fatty acids in the liver, the normal regular people is not from de novo lipogenesis, but when people get insulin resistant, this contribution is very important. At least 30% of the fatty acid pool in the liver of these people is from de novo lipogenesis. This animal model seems to be 100%, at least. SRBP1C is not required for IPAC2 mice hypertriglyceridemia or fatty liver, which is interesting because in our general model, exaggerated levels of insulin are the main driver of fatty liver and hypertriglyceridemia in the metabolic syndrome context. We don't know if this is true in our lipodystrophic patients or in the general population of people with metabolic syndrome, but at least suggests that ChREBP can be very important too, yeah? And eventually say a pharmacological target to tackle these complications in humans. So mechanistically, because everything I have shown at this point is more descriptive, we are facing a paradox in which obesity and lipodystrophy weight apparently the same, even though these are heavy people and this is very lean people, but they weigh the same from the metabolic point of view. Both conditions cause insulin resistance, the excess of fat and the lack of fat. This is a paradox, or maybe it's an apparent paradox, because we have no answer for at this point to understand this thing. However, we have at least two very good hypotheses. I said they're very good because there is experimental evidence supporting both of them. As showed Mariana yesterday, the adipose tissue can work as a metabolic buffer in the sense that adipocytes prevent the lipotoxic buildup in several tissues that are key to preserve insulin sensitivity in the whole body. The second hypothesis that can explain this paradox is that the adipose tissue is also an endocrine organ. Also, this was explained yesterday by Mariana. So adipocytes secrete a lot of hormones, classical hormones like proteins with biological activity, but also lipids that can act as a signaling molecules in distant tissues. Of these ones, obviously leptin seems to be the most important, or at least the best known one, or at least the most studied one. Adipose tissue essentially prevent the excess of circulating fatty acid after we eat fatty food. So they store this excess of fatty acids after each meal in the form of triglycerides, and they release these molecules in a very regulated way after during the fasting period. So you can conceptualize, imagine the adipose tissue as a container. So this is the normal situation that you see in normal people, or eventually in this subset of obese people that have no insulin resistance, the so-called metabolically healthy obese people. So normally adipose tissue expands, and actually this is happening every single day. Actually, in this very same moment, our adipose tissue is getting shrinked because we're not eating, right? So we're using the energy that is storing out our adipose tissue. But then, in our next meal, adipose tissue will be expand a little bit, and this cycle is normal. But this normally expanded adipose tissue prevent the spillover of lipotoxic lipids into these critical tissues. So in this theory, when insulin resistance appears, it happens that adipose tissue cannot be expanded normally. Yeah? This abnormal expansion can be either quantitative or qualitative, as Mariana explained yesterday. It's kind of paradoxical that you can say, well, if this very severe obese patient have an insufficient expansion of the adipose tissue, but eventually for the flux, the total flux of fatty acids in this person is still insufficient. When this happens, muscle, liver, and pancreas, and maybe other tissues, including some areas of the brain, can be overloaded with these lipotoxic lipids, which are not really well understood which ones are those. Which is clear is those lipotoxic lipids are not triglycerides, because they're very unreactive species from the chemical point of view. In lipodystrophy, you have the extreme situation. You have no adipose tissue, and when lipids are getting into the system, they cannot be stored in anywhere, and they simply get uptaken by the muscle, liver, and the pancreas, and they express under these circumstances the maximal lipotoxic insulin resistance. This is just to illustrate this point. One of these experiments supporting this adipose tissue model as a buffer, as a metabolic buffer. These are the OB mice that I already mentioned. These mice are morbidly obese. They lack leptin, so they have hyperphagia and develop diabetes, insulin resistance, and hypertriglyceridemia and fatty liver. These animals are OB plus the transgenic overexpression of adiponectin in the adipose tissue. So these are real super obese animals. You know? They're such obese that they die because when they get upside down with the feet facing the sky, they cannot move back to the normal position and die because they starve to death. So that's apparently the cause of death of these animals. Normally, a mice weighs between 20 and 30 grams. During these weeks of observation, you can see how the regular OB mice can weight up to 40 or 50 grams, meaning 2-3 times the normal weight. But these super obese adiponectin transgenic mice can weight up to 80 grams. Yeah? Curiously, these super obese ones have no metabolic abnormality whatsoever. If you can compare with the normal values here, and glucose is normal, insulin is normal, triglycerides in plasma and free fatty acids are normal compared to the regular OB mice. At the adipose tissue level, the OB mice normally have these very large adipocytes with presence of cells and expanded extracellular matrix in between, and also inflammatory cells here. And the OB mice plus the overexpression of adiponectin in the adipose tissue have a normal size adipocytes with no expansion of the extracellular matrix and inflammatory infiltration of the adipose tissue, indicating that independently of the expansion of the adipose tissue in terms of the mass, it's very important the quality of this adipose tissue. So this data is supporting the idea that adipose tissue is important for storing and preventing the spillover of fatty acids into the circulation and then to these other tissues. So in conclusion, the adipose tissue, I didn't talk about this, but it was probably evolved in, as an adaptation to prolonged fasting, but was acquiring additional regulatory functions during the evolution, including the secretion of these hormones in order to coordinate the whole body metabolism. Systemic insulin resistant results from abnormal adipose tissue remodeling, as I already showed you with this data in the super OB mice. And lipodystrophy is a rare disease, but it's a natural case of extreme adipose tissue dysfunction. And we believe that the study of this condition can highlight aspects of adipose tissue physiology, and but also novel pathophysiological ideas or mechanisms for developing new therapies against more common forms of insulin resistance and eventually also for obesity. That's this. Thank you very much again, Rodrigo and the organizers. A very nice meeting. Hopefully it will be going on this thing under the directions of I don't know who. You haven't said who's your successor, but thanks again.
Thank you very much, Victor. So we have time for only 2 questions. So the next successor.
Thanks, Victor. Wonderful presentation. One question. The patients are hypertensive according to the medication that they receive.
Correct.
Is it because elevated glucocorticoids? And also, what about the knockout animals? Are they hypertensive too?
Well, yes. Hypertension is a very common disorder. Yep. I don't know if these are very few patients that have been described in the world. Okay. But makes sense that, as you mentioned, the cortisolemia eventually is driving this thing, but also might be a different causes. For example, I didn't mention, but these patients develop kidney failure. That can be another potential explanation. Also, insulin resistance is a cause of hypertension. So there are so many different possibilities from the pathophysiological point of view for hypertension.
Thank you for your talk. I was thinking in the skeletal muscle in these patients. How is the quality of the muscle because of all the stress that the body is experiencing?
Correct. We haven't studied skeletal muscle in the patients, but in the mice, muscle is decreased. Yeah, it's very decreased. And again, it can be a consequence of this hypermetabolic status, but also for as a complication of the hypercortisolemia. It can be an explanation too. The excess of cortisol is a catabolic signaling for the muscle.
Thank you very much, Dr. Cortez. So we give him an applause again for his great work. So our next speaker is Roberto Bravo. Roberto received his PhD degree in biochemistry from the University of Chile. Currently, he is assistant professor at the INTA from the same university. Dr. Bravo's research focuses on studying the communication between the endoplasmic reticulum network and the mitochondrial network. He has demonstrated that this mechanism is important for several pathological processes, including insulin resistance, cardiovascular diseases, and obesity. More recently, Dr. Bravo's research has begun to show how this communication participates in cellular senescence. So we invite Dr. Bravos to this podium to show his research on pre-adipocyte-derived inflammatory secretion as a driver of hepatocyte cell senescence. Thank you very much.
Thank you for the invitation. So, as Professor said, I'm from INTA. I'm from Laboratory Omega, just like Mariana Cifuentes. So we share the same laboratory. And I'm also part of ACDIS, the center that Professor Andrew Quest presented yesterday. He was in fact my PhD thesis director. And I'm also part of the CS, which is the Inter-University Center for Healthy Aging, which is based here in the University of Talca. So this is not my first time here. And I'm an aging enthusiast. So we're gonna switch from obesity to talk about a little about aging, or talk about aging from the point of view of obesity. We're going to talk about the aging process, cell senescence, adipaging, which is a term I found in the literature, which explains how obesity accelerates aging, which is a spoiler from the talk, and also the final part, which is I'll show you a tiny bit of the result we have in this topic, in which adipose cells drive hepatocyte cell aging. And also in this right side of the slide, you'll see a progressive summary, where you find the key concept of each slide. So, as a prologue, why is this important to study aging? It is because Chile is one of the countries with the fastest aging paces in the world. This is our population pyramid here. From the census, we have around 17% of older people, and the projection, which is similar to what Gerardo showed yesterday, is that by 2050 we'll have around 1/3 of older people in our country. So this poses a lot of challenges we have to tackle, and we need to understand the aging process to address that. And also, older people face different challenges. There are changes in many different dimensions. For example, like work and pensions, dependency and autonomy, family and cares. But I also wanted to talk about 2 words about discrimination, which the discrimination because of age is called ageism, and older people experiment discrimination because of the older age, which characterizes as stereotypes, prejudices, and discriminatory actions and behaviors which we have to eradicate from our culture. For example, there are micro-discriminations. For example, youth is the prime of life. In Spanish, la juventud es la flor de la vida, which is a very nice thought. However, this is discriminatory because each age is different. So each age can be the prime of life. So let's talk about the aging process. The aging process starts even before we are born, because there are genetic factors that determine how we age. Also, the environment and our lifestyle choices determine the course of our lives. Once we reach our adult age, then from then on, we experiment the aging process, which corresponds to progressive decay in the tissue functions. This is due to an accumulative damage, and we all age differently, because when we reach old age, we can become a robust older adult or a frail older adult, depending on how we reach our this stage. So now we have 2 different concepts about aging. We can have chronological or biological aging. In this example, I show you the face of a British truck driver. And as you see, in one side of his head and of his face, we see the characteristics of aging, like wrinkles and changes in fat composition of the face. But in the other side of his face, we can see more profound features of aging, like more intense wrinkles. This is because he was a truck driver. The one side of his face was always facing the sun, so he was exposed to UV radiation on one side of his face more frequently. So this led to cumulative damage that accelerated the aging process. And thus, aging corresponds to changes in many different aspects. Now we know about 12 different hallmarks of aging that change throughout our lives due to this cumulative damage. The most important, for me at least, the most important hallmark of aging is chronic inflammation, which also characterizes by changes in endocrine saturations, metabolic imbalance, dysbiosis, and a lot of other changes that, due to time constraints, I cannot address it right now. So, in the aging process, we know that during aging there's a deregulated nutrient sensing. For example, we know that calorie restriction is one of the interventions that prolongs lifespan or health span, depending on the species. But on the other hand, we know that during aging, there are changes in this metabolic balance, in which the abundancy balance, the abundancy signals, for example, like insulin, they are increased, and this also drives the aging process. There's also this concept metaflammation, which is the combination of the words metabolic and inflammation, which means that changes, alterations in the metabolic state leads to inflammation. For example, obesity, which is an alteration in the adipose tissue, as Mariana told yesterday. It induces inflammation, and also it is known that the inflammation due to obesity leads to metabolic alteration in other organs, such as, for example, how the obesity, which is this excess in the adipose tissue, leads to metabolic alterations in the liver, for example. Also, aging characterizes by changes in endocrine factors. For example, interleukin-6 levels are increased throughout our lives, and which is a pro-inflammatory cytokine, which also contributes to the chronic inflammation. Aging also leads to dysbiosis, which Gonzalo talked about yesterday also. Changes, which are changes in the different microbes in our gut. Also, we have accumulation of senescent cells, which will be the core of what I will talk about in the rest of this lecture, in which accumulation of senescent cells also contributes to chronic inflammation, because senescent cells acquire a senescent-associated secretory phenotype, SASP, in which they secrete a lot of pro-inflammatory cytokines. And also these all affect stem cells, which affect regeneration through, due to, which decreases while we age. So, about cell senescence, what it is. When we have a tissue that experiments damage, if this damage is too low, the cells can adapt to it, so they can restore homeostasis by their own. If the damage is too high, it induces cell death. But however, when the damage is medium, cells become senescent, which means they stop proliferating as a means to stop the propagation of the damage to the daughter cells. They also secrete pro-inflammatory cytokines, which recruits cells of the immune system, like macrophages. Macrophages remove the senescent cells, which leads then to a regeneration process. So, cell senescence is a pro-inflammatory, or say is an inflammatory process. Inflammation is a physiological process which is required because when inflammation switches off, it leads to regeneration. However, during aging, there's an accumulation of senescent cells. This is because of our immune system with age becomes less efficient to remove the senescent cells. So senescent cells accumulate in the tissues during aging, which helps to build up the chronic inflammation that I told you before. So, what are the features of senescent cells? As I told you, cell senescence can be triggered by damage, also by deficient replications, for example, telomere shortening, telomere attrition, and also activation of oncogenes that also can trigger this process. this leads to the activation of a classical cell-senescent cascade, which is P16, P53, and P21, which lead to cell cycle arrest. Also, it leads to the activation of a pro-inflammatory cascade that leads to the, as I told you, the secretion of pro-inflammatory cytokines. This is important because pro-inflammatory cytokines act on their receptors on the cell surface, and they can act on the same senescent cell, so sustaining a positive feedback loop that maintains the pro-inflammatory state. Also, these pro-inflammatory cytokines can have a paracrine effect, so they can affect neighbor cells, leading to the propagation of the inflammation to these neighbor cells. And it is known that this inflammatory pathway also leads to cell senescence. So this means that that is the importance to remove the senescent cells, because if you do not remove the senescent cells, the pro-inflammatory cytokines build up in the tissues and drive cell senescence in the neighbor cells. They are also resistant to apoptosis because of the activation of anti-apoptotic pathways. And this is also related to metabolic alterations. As I told you, the different hallmarks of aging are intertwined. This is why, for example, hyperinsulinemia activates growth factor cell signaling that has an anti-apoptotic effect. So metabolic defects, metabolic imbalance, which leads, for example, to hyperinsulinemia due to insulin resistance, also contribute to the development of senescent cells. So colloquially, senescent cells are also termed zombie cells because they do not work properly, they are barely alive, they refuse to die, and also they can infect other cells due to the pro-inflammatory signal. And this leads us to the adipaging concept, which is the combination of the words adipose and aging, because it is known that obesity accelerates the aging process. Because as Mariana told you, she's like the obesity queen here. She said everything yesterday. We have that during obesity, this metabolic unhealthy obesity, there's an increase in adipocyte size, which leads to the recruitment of macrophages, which also leads to a pro-inflammatory state in the adipose tissue, which also leads to a pro-inflammatory state, a chronic inflammatory state in the whole body due to the secretion of pro-inflammatory cytokines from the adipose tissue. So this process that I told you that occurs during obesity is very similar to that of cell senescence, because both include the recruitment of cells of the immune system and the pro-inflammatory state. The difference is that in cell senescence there is damage that causes cell senescence, which is typically genotoxic stress. However, in this case, this is not the triggering of cell senescence per se, but there's an metabolic stress that leads to the pro-inflammatory state. And thus, obesity shares many of the hallmarks of aging, like chronic inflammation, which is obvious from the secretion of pro-inflammatory cytokines. The metabolic imbalance that we see during aging is also exacerbated during obesity. This biosis that Gonzalo talked about yesterday that happens during obesity is the same process, is the same alteration that occurs during aging, and there are other features that we cannot talk about right now due to time constraints. So, what we know about the different diseases related to obesity is that, for example, in lessons from animals, we know that when they are fed with a high-fat diet, they become obese with an inflamed adipose tissue, which leads to the secretion of pro-inflammatory cytokines that affect other organs, for example, the liver, which accumulates fat in this disease called fatty liver. So, knowing this process that the inflamed adipose tissue can lead to disease, we wondered whether cell senescence is part of this mechanism. Can the inflamed adipose cells lead to cell senescence in liver cells that would contribute to the disease? So in this work, we used a model that Mariana set many years ago, in which we can use a cell line, an adipose cell line. In this case, LS14 cell line, which is a pre-adipocyte cell line that we can also differentiate to adipocytes. When these cells are treated with cinacalcet, we see there's an increase in pro-inflammatory cytokines like interleukin-6, CCL2, interleukin-1 beta, in both pre-adipocytes and adipocytes. What is cinacalcet? This agent that induces inflammation in these cells is an activator of the calcium sensing receptor. Upon activation of this receptor, it leads to the secretion of pro-inflammatory cytokines from the cells, which leads to a pro-inflammatory state in the same adipose cells, which further induces the expression of the calcium sensing receptor. So activation of this receptor in adipose cells leads to a positive feedback loop of inflammation, which serves as a model of inflamed adipose cells. So now, a tiny bit of what we did with that model is this is a little long title, which I hope it will be clearer at the end of the presentation. What we did with Mariana, we co-directed the PhD thesis of Lautaro Briones Suarez. He is now a doctor. We already published these results. We used another cell line of pre-adipocytes, which is the SW872. And these cells were treated with either vehicle or cinacalcet for 16 hours. So these are control cells that are not inflamed. These are cells that have been that are inflamed. And then we changed the medium from fresh medium. So we have a conditioned medium from cells treated with vehicle or with cinacalcet. Importantly, as I told you, here there's a medium change. So cinacalcet was present in this first medium that led to adipose cells inflammation, but is not present here. This conditioned medium has the secretions of the inflamed adipose cells. And then what we wanted to do in this in vitro model was to use the conditioned medium from the adipose cells and use them on HepG2 cells, which is a hepatocarcinoma cell line, which are cells that in vitro behave very similar to hepatocytes. So we wanted to know if these if this medium can has an effect on the development of cell senescence in these HepG2 cells. So first of all, we measured the expression of senescence-associated beta-galactosidase, which is a signature of cell senescence. And I don't know if you can see it from where you're sitting, but we have here the cells that were exposed to vehicle-treated cells, that were exposed to the conditioned medium from vehicle-treated cells, so these are the control cells. And these are the cells treated with the conditioned medium from inflamed adipose cells. So we see here that there's an increase in the blue color, there's an increase in the beta-galactosidase positive cells, not only the positive cells, but also the color, the absorbance of this dye. It increases, indicating that indeed, as we expected, the secretion from the inflamed adipose cells leads to the accumulation of senescent cells in our experiment. We wanted to know if this is indeed due to the secretion of pro-inflammatory cytokines. So what we did was perform again the same experiment, but we also immunoprecipitated some pro-inflammatory pro-inflammatory cytokines. For example, we see here this is the count of senescent cells with the conditioned medium of the cinacalcet treated cells. So we have here the maximum amount of senescent cells. Then we in this medium we immunoprecipitated interleukin-6. We did not see a significant change. But when we immunoprecipitated interleukin-1 beta or TNF-alpha, we saw that there's a decrease in the number of senescent cells, meaning that these pro-inflammatory cytokines from the conditioned medium from the inflamed adipose cells is responsible for this increase in the senescent cells. Of course, we wanted to know to measure other the other features of senescent cells. We can't only have one marker. We need several of them to tell that those are indeed senescent cells. So we measured the cell signaling cascade of cell senescence, and we see that the conditioned medium from cinacalcet treated cells indeed increased the expression of P21 and P16. We also wanted to know if this indeed leads to a cell cycle arrest. So we measured the nuclear levels of Ki67. This is a protein that is located in the cell nucleus during cell proliferation, but when there's a cell cycle arrest, its nuclear levels decrease. So we have here the control cells in which we have a strong Ki67 expression, but with the conditioned medium from cinacalcet treated cells, we see that there's a decrease in Ki67. So this is indicative of a cell cycle arrest. And finally, we also measure the expression of pro-inflammatory cytokines. We have here the expression of the mRNA of interleukin-1 beta and CCL2, which are pro-inflammatory cytokines with part of the signature of cell senescence of HepG2 cells. It is important to note that this is the mRNA from these cells, so these are not cytokines from this medium, but are the production of mRNA of pro-inflammatory cytokines in the cells. So we have here that there's an increase compared to control cells of both about pro-inflammatory cytokines, meaning that we have indeed not only the common features of cell senescence, but also the induction of SASP. So in conclusion, we have here that indeed when we activate the calcium sensing receptor in SW872 cells, they become inflamed, they secrete pro-inflammatory factors that when we expose HepG2 cells to these pro-inflammatory factors, they become senescent. So this is a potential mechanism by which obesity, metabolic unhealthy obesity, might lead to this pro-inflammatory state that leads to alteration in liver cells. So this is going back to the one of the first slides I showed you. This is very similar to what happens with the difference between chronological aging and biological aging. If you're a truck driver and you do not use sunblock, you'll have in one side of your face the increased features of aging, and on the other hand, if we have altered metabolic state, for example, obesity, this also contributes to an accelerated aging due to this effect on the buildup of senescent cells in other tissues, for example, the liver. So thank you very much for your attention.
Thank you very much, Roberto. So I don't know if there are some questions. Okay.
Thank you very much for the presentation. I have a question mostly on observation. It's completely partial observation. And I'm going to give an example of my father. When my father was younger, he was a little bit obese, and he looked older. And now that he's really old, he is really thin, and it looks older than his age. So, is it possible that at some point, for example, when you're younger, obesity makes you look older, when you're older, obesity makes looks you younger? So is it possible that obesity in some point of age can have a little bit of protection against aging or senescence? Or is it possible? Just basic partial observation.
Okay. Well, one thing is the aging process, biological aging, and the state of our body, and the other is how we look, which is different. So if the part of our looks can depend on the skin wrinkling and the fat composition. So I cannot address that because I'm not an expert in the aesthetics of aging, but I can tell you that on the one hand, obesity accelerates aging from a metabolic perspective, but at old age, it is known that overweight people have a better survival chance compared to normal weight people, which is a paradox, because I'm telling you obesity and overweight accelerate aging, but it is protective at old age. And this is one hypothesis, is because when you are overweight, your muscles have more work to make you move. So that means that if you're overweight during your adulthood, you're exercising for your muscles, so you have better muscles when you are older. So that might be part of the explanation on that, However, I don't know your father, so maybe there are a lot more to that.
So I have a question regarding your model, because you show that pre-adipocyte treatment with this drug induces a pro-inflammatory state, right? But could it be that these cells, these pre-adipocytes and adipocytes, are really senescing? I mean, in response to this drug, and what you are seeing really is the pro-inflammatory response of an accumulation of senescent adipocytes there that in turn can secrete things that impinge on hepatic cells. So what, in other words, what you are seeing is two senescent events in your model. And this is because, I asked you this because there are some studies showing that people with, or animals with metabolic syndrome, for example, if you take a biopsy of the fat, you see an accumulation of senescent adipocyte cells. So it could be that what you are inducing really at the first is senescence in adipocytes. Huh?
Yes. Cell senescence is this process in which cells stop proliferating to avoid the propagation of damage. That means that terminally differentiated cells, they do not proliferate, so they cannot stop their cycle. And on the other hand, the non-differentiated cells, they can proliferate, so they can stop their cycle. So we have a difference that non-differentiated cells can undergo cell senescence, but differentiated cells do not undergo cell senescence per se, but a senescent-like phenotype. Now, if we think about the adipose tissue, we have the adipocytes, which are differentiated cells, and we have the pre-adipocytes, which are the undifferentiated cells that proliferate. We perform this research in pre-adipocytes, which are the ones that can proliferate, and then they can undergo cell senescence. That's why what we see here might be a communication from one senescent cell to another. So we did not address that because we used it as a model of inflammation and not as a model of senescence. But of course, we can measure that, and I'm a senescence freak. I'm a fan of this process, so I suppose I see senescence everywhere, so I suppose it is possible that these cells are senescent.
Yes, it's interesting because, I know what you mean, but some people also claim that terminally differentiated cells show some features of replicative senescence in a sense. For example, the accumulation of beta-galactosidase at low pH, etc. So it could be that even when those cells are terminally differentiated, and once you treat them with the drug, they can express some features of senescent cells.
Yes, exactly.
Any further questions?
Thank you, Roberto. So you talk about this chronological versus biological senescence, but I wonder if every single cell in our body have the same biological age. For example, how you can define the biological age of an organism if your type A cells have one age and the type B or type C cells have a different age. So you average everything or what?
No. I showed this slide, but I did not talk about any of these. This is the one. The 12 hallmarks of aging. you can measure them in different cells, different organs, but different cells behave differently. For example, one of the hallmarks of aging is genomic instability, which means the mutation rate in the different cells. We know that is different, for example, in the mutation rate in cells that do not proliferate. For example, neurons is very different from the mutation rate in epithelial cells, for example. So from only the point of view of this hallmark of aging, we can say that all the cells in our body have different aging clocks that are not the same at all. And if you ask me about what happens with obesity, is that it is known to increase all these hallmarks of aging, and in some point of view, obesity accelerates the aging, and to say something, it synchronizes the clocks of the aging cells, because we should have cells that last longer, but with obesity we have the increased rate of aging. So if that is what happens with the different components, but you ask me what happens as a whole, do we average everything? And I cannot answer that, because we are complex organisms. So I can say, well, I can be 50 years old here and 25 here, 2 years old here. So I don't think there's one age, one biological age in our bodies.
Okay, thank you very much, Roberto, for that very interesting talk. So, last but not least, yes. So, so the last speaker is Flavio Contreras Torres. Dr. Contreras Torres received his Doctor in Science degree from la Universidad Nacional Autónoma de México. He completed his doctoral studies in chemistry at the Institute of Nuclear Sciences, working under the guidance of Professor Dr. Vladimir Basiuk. Following his PhD completion, Dr. Contreras Torres was awarded several prestigious fellowships and postdoctoral, especially a postdoctoral fellowship program. In 2016, Dr. Contreras Torres joined the School of Science and Engineering at Tecnológico de Monterrey as a professor and researcher. Over the years, he has made significant contributions to the scientific community with a total of 60 refereed articles, 3 book chapters, and numerous conference presentations. Dr. Contreras Torres is member of the National System of Researchers, the American Chemical Society, and the International Union of Crystallography. In 2022, Dr. Contreras Torres assumed finally his role of a core researcher at the Unit of Experimental Medicine at the Institute of Obesity and Obesity Research at the Tecnológico de Monterrey. Currently, his research interests revolve around the application of theoretical and experimental approaches aim at developing fundamental understanding of the interactions between obesity-related receptors and their ligands. So without further ado, we invite Dr. Flavio Contreras to talk about exploring computational approaches for targeted target-based drug discovery within obesity pathways. So thank you very much for joining us.
Thank you for the introduction. Thank you to everyone for being here. It's a pleasure. My first stay here in Chile. I'll be very happy with your attendant, Dr. Rodrigo Moore, and to everyone. So I hope this talk today will show you, will be also by your interest. My talk today is about a different aspect, as I think the only one, but there is one student for over there is you and me, the two ones who study with theoretical the obesity, but using theoretical approaches. So my talk today is about these tools, theoretical tools. Okay, so I'm going to continue. Here. Okay. Obesity is a complex condition which affect in different ways to men or to women, as we can see from a good paper from the Lancet journal. We can see the rate of the obesity increase over the years, and it is more observed in women than men along the years so far. The obesity, there is very interest to this condition, including as other people talked today or yesterday about genes, about sedentarism, about the diet, and also about the drugs, for example, antibiotics, as yesterday Dr. Chamorro, I think, talks about the good microbiota can be changed by using some drugs. The obesity also has very effects, including cancer, as Professor Andrew talked about today, cardiovascular effects, pathologies on the digestive system, and also going to the depressions, but they are we have to understand the obesity as a very conditions which have to be connected, each one, but currently we do know how they are connected. So our approach is to study the obesity in different organs as well as with different connections doing axis between the organs. For example, with the cardiovascular diseases, we know diabetes is a condition to going for cardiovascular effects, but also other issues as, for example, the obesity. And the axis we are interested in study in our institute is the axis with the heart, with the liver, with the adipocyte tissue, and the obesity has also very treatments for obesity, very approaches to the treatments. One of them is the surgery, but not everyone is conditioned to the surgeries. Other treatment could be the exercise, other treatment could be the nutrition, but also pharmacology. And my talk is more focused on drugs and novel drugs, which could be the new drugs for obesity. The obesity market around the world is very is increasing in these years, and it is expected to reach $50 billion by the next years. So the market for the pharmacology for obesity is rising every year. Okay, in my department, or unit as we call it, we have the approaches to look for obesity treatments or understanding the obesity in order to go forward translation. This means we need treatments for population, for people, okay? So the unit is, there are several lines of research in this unit, and the mine is the first one, the in silico approaches, because we need to predict, we need to propose mechanisms which can be proved on cell models, think forward to tissues, in 3D tissues on immune cells, animals, models, to go forward to the people. So my talk is about only, I don't know the point there. Which is the pointer? Can you hit my, please? Okay. Okay, thank you very much. So my laboratory is, is this one? Okay. It's called nano and microstructures. And we, as I tell you, we are study pathologies of obesity doing models, theoretical models. The global pharmaceutical industry in obesity is studying the obesity as mechanisms. And the four main mechanisms is about the appetite and satiation regulations, the energy expenditure, the lipid metabolism, and the good microbiota and satiety. And several companies around the world, they are studying the obesity according to these four mechanisms. The main one is the good hormones and satiety mechanisms, and we have the main company in working on pharmacology for obesity, which is the Novo Nordisk, and they are studying glucagon receptors for obesity, but the other ones are looking for obesity, studying lipid mechanisms as Roche or AstraZeneca for mechanisms related with satiation regulation. Here, there is important to have these two words. They are not the same, satiation and satiety. Satiation it's understood as a condition, a mental condition, and satiety it's related with the sensation of I eat well, okay? The only 6 drugs which are approved by the FDA They are those ones: lorcaserin, liraglutide, orlistat, phentermine, topiramate, and naltrexone. And not they are the best one. They are working only for specific mechanisms. Along the years, they were other drugs for obesity, and most of them were withdrawn because they show safety concerns. Okay, and the pharmacology of obesity is very, very, very clear that wherever the drugs could be, they is very probably they show safety concerns. So this is changing every year, and several papers are reported proposing novel drugs or proposing novel molecules for obesity. So we need the testing on living cells for say something. Okay, but what's the main problem on the drug discovery? The drug discovery is comes from two paradigms. And in translation point of view, we need testing the treatment in the populations, and our models go forward with animal, cellular, or molecular targets. But it's not happening in drug discovery, because in drug discovery, much drugs we are today using, they come from serendipitous observations. One of them is, in mine is, for example, I'm sorry, I forgot the names of some drugs which you are using, but not for this translational focus on research. Okay, in a recent paper, in a recent kind of review paper, it is observed, it is discussed how the target-based drug discovery were increasing along the years, but comparing with phenotype drug discovery, it is very, very small. Okay, actually, the drug discovery starts in 1995 with HIV, because also the computers, the modern computers, and the software arrived in this year. To our days, we have very powerful computers, so we can model systems that before this year were impossible to do. Okay, so the target drug discovery is quite new in the field of drug discovery. Okay, so which is our approach, in silico approach? My talk will continue with this approach. In my laboratory and in your unit and in our institute, we are using cheminformatics tools. We are using machine learning tools, as well as biophysics tools. Those tools are not new. Cheminformatics is using before the 18th. Machine learning were developed in the 40s. And biophysics is quite old also. But in the context of the drug discovery, they are they are using, in the context of biomedicine, and they are proving they can be useful tools for also study very complex diseases such as obesity. Okay, so my talks will be focused in these examples for pathways related with obesity. Of course, as you attending in this summer school, they are very much receptors which can study. Okay, so it will be very hard to explain all of them. And also, I want to tell you this line of research is quite new. Is almost 1 year and a half. So we have to build the computers or workstations. So the facilities were also new as the line of research. So this is the time we are studying this receptors is very new. So the results I'm going to show you are under study. They are pre-data if you want to understand as this. But I expect in the next years be here also, but with the final results. So it is like a lecture. It is not at my presentation is not as the final results. Okay. So I will focus on the PPARs. I will focus on cannabidiol receptors, and I focus on TL receptor. Okay. In my first example, which is actually was led by Professor Gerardo Garcia, and this is under revision today, in these days. We study the PPAR gamma because PPAR gamma is a receptor which is involved in adipogenesis. Okay, so the PPAR gamma, they are the PPARs, the family of PPARs are located in the nucleus, and they have these proteins. They have these structures in which they are about 12 helices. They are 4 or 5 beta sheets, and they are one loop, which is called serine 2, 4, 5, in which occur the phosphorylations of the protein, and because of phosphorylation, the transduction of some genes change. Okay, so the orthosteric volume of PPAR gamma is this region in yellow, and all drugs that may be agonists or may be antagonists with PPAR gamma may be located in these regions, because the amino acids interact with the proteins, with the drugs, and if the interaction occurs with the hélice H12, the hélice move, okay? And this movement changes the codification of the genes, okay? So if we are looking for agonists, we need drugs that interact precisely with H12. The other drugs which also interact with PPARs, or PPAR gamma in this case, also interact in the orthosteric volume, but they are not interacting with H12, but they can interact with, for example, H3, the allele H3 or the allele H5 or other regions. Some researchers I proposing the serine 245 could be a better point to interact with this protein in order to change the to change the translations of the genes. So, but this is just an hypothesis, and the pharmacology are more involved in how my molecules can interact with the H12. Okay, the drug which interact with PPAR is rosiglitazone, but rosiglitazone, as I say, have many safety concerns, so it was withdrawn of the pharmacology of the, and this is used now for just only in vitro studies or in silico studies. When the PPAR gamma in this case modify the H12 allele, the adipocytes, the mechanism for adipogenesis, changes. So the fatty acids going forward to the inside the adipocytes, and the cells require now glucose. So the mechanism is involved now, dependent on glucose. So PPARs are mainly focused with is a molecular target for diabetes. Okay, because they change the mechanism and it is more dependent on glucose. Okay, so if we have one drug, we can modify this PPAR, this receptor, by interacting with the H12. We have an full agonist, and there are several molecules which are classified as full agonists. This family is the glitazones, okay, and they are proving, but they are not proved, because as a family they also has several safety concerns. So we are thinking which could be a better drug as rosiglitazone that can interact with PPARs, but without the safety concerns. And in our laboratory, we are very focused on the use of cannabidiol. So we study not for obesity, but for other objectives. But we use CBD. Experimentally, it was measured the expression of CBD in the PPARs, but it is not my focus in this talk. And we observed in silico that cannabidiol also interact with with PPARs gamma, and the energetic analysis determines that cannabidiol may compete with rosiglitazone as an agonist. Or a study were carried on using molecular dynamics, 500 nanoseconds, 5 replics, and using these potentials, and main conclusion of this in silico results is that CBD may compete with rosiglitazone. So we have another option to the glitazones families. Okay. Okay, but the PPARs, the PPAR gamma, the PPARs are a family. So we have the PPARs alpha, the PPARs delta, or the PPARs beta, and they are like a siblings. One sibling say something, the brother say something else differently. Okay. Meanwhile, PPAR alpha is related with fat oxidations, the PPAR delta is related with fat burning. The PPAR alpha is expressed in liver, and the PPAR delta is expressed in several tissues. So we carry out molecular dynamics studies in order to observe or to predict the interactions of cannabidiol in these PPARs, and we observe by our studies, in silico studies, that the CBD preferentially can be bounding more preferentially in PPAR delta than the PPAR gamma. So what happened if we are studying or we are in a real case, we are attending a patient with cannabidiol for diabetes with cannabidiol. So we may think that the drug can interact with all of the families of the same protein because they evolutioned with the as a family. Okay. So we have rosiglitazones, we have the glitazones, we have the cannabidiol. But our main question is, there could be other molecules in order to act as an agonist, antagonist of PPAR gamma. The answer is okay. You look for them. Okay. So how was our approach in order to search these molecules? Okay. We started doing PC analysis, and we use a database, which is the PDBA, the protein data bank. In the protein data bank, they are the crystals of any molecule who were crystallized with the PPAR. In this case, the database contains 244 entries. Okay, doing the PC's analysis, we were able to cluster in the different molecules and understand how can be these molecules. As a LIGO, the pieces which we use in order to construct something as a LIGO, these are the parts of the molecules. Okay, so we can see if I would like to find agonist or antagonist of PPAR gamma, my new molecules, my novel molecules, maybe could be structured by these LIGOs. Okay. This is not the full history. This is the beginning of the history. Okay. Where could be my molecules which show these parts? Okay. Okay. So going to other database, for example, Coconut. And why Coconut? Because Coconut is a database of natural compounds. This database has more than 40,000 entries, so there can be some molecules which can act as a PPAR agonist. Okay, so we filtered this database in order to look 1 in a million molecules. Okay, so how we can do this? It's doing a PC analysis is very impossible. We can obtain nothing doing a PC analysis in a very large database. So we classified we did another type of analysis which clusters by families, and we categorize this database, this chemical space, in families as catechins, anthocyanins, capsaicins, proanthocyanins. Okay. Then I'm going to talk about this. it is in this precise time. I'm not going to talk about machine learning, but doing machine learning with these families, we obtain about 41 new molecules, probably as agonists for PPARs. Okay. With this 40 molecules, we did a virtual screening study in which we obtained the border pose of the better pose of the molecules into the orthosteric site. And with the energy of these interactions, we can choose the strongest interactions. Okay, obtaining these interactions, we perform with other filters, which could be stable along the time by doing molecular dynamics. We obtain new, probably new nine natural products, which could be acting as an agonist of papers. Okay, doing the molecular dynamics and performing the analysis, by the binding energy and the interactions. So we obtain these this distribution of binding energies as comparing with the rosiglitazone and CBD, and we observe that this catechin, interacts more stronger than the other ones. So, what happened? Okay, let's do a more large molecular dynamics study. Okay, which is more large? It's about 500 nanoseconds, 3 replics, and we obtain finally the stability of the new molecule, in this case, the capsaicin, in the pocket of the PPAR gamma. Okay, so we study which are the main interactions of this molecule inside of the PPAR gamma orthosteric site. So we observe that they are interacting with several amino acids, and they are interacting also with two bonding, H bonding. But the main result is they did not change, this catechin didn't change the helix 12. So, as I tell you, it cannot be considered as a full agonist for PPARs. Okay, so these are the main interactions. As you can say, they are the distance of the bondings, they are the interactions type of the catechin inside the PPARs. You can say many H bonding interactions as well as hydrophobic interactions. Okay. Going forward to the other probably potential molecule, proanthocyanins, we observe now that as a main result, along the time of the molecular dynamic simulations, the helix H12 changed their position. So this result is telling us they can, as PPARs change the orientation of the helix 12, they probably can act at the gene level, okay, with another transduction than the spec. Those are the interactions, H bonding, hydrophilic, and the last one, the capsaicin, was the better one because they change hardly the H12, which is related with the transcription of the PPARs, and the H bondings are stable along the simulations, and capsaicins, in this case, this capsaicin interact with PPAR in this amino acid in cysteine 2A5, which is a very no point of interaction as rosiglitazones can do. So the capsaicin is proposed as a full, this I think the interaction, the capsaicin as proposed as a full agonist. So we finally discover or propose or predict another molecule for piperam. Okay, in these pictures you can say the ligos of the new particle. Okay, we predicted these ligos should be on whatever molecule we predict. So they are in these capsaicins. Okay, the final analysis is an ADMET analysis, and we can see it complying with by comparing with the others, with the other natural products, catechin, proanthocyanins, and rosiglitazones, and cannabidiol. We can say we can propose the proanthocyanin and the capsaicins as a full agonist. They are predicted. Of course, we need to do the experimental validations, and they are they have no violation of the Linsky rules, but CBD CBD actually do the violation of these rules. Okay. Okay, my second example is the cannabidiol receptors. In this example, I'm going to tell you about the machine learning tools. In this example, we are looking for agonists, antagonists for these receptors, but using machine learning tools. So in machine learning, we carry out two main ways. The first one is the training of the algorithm. The training needs to be very smart in order to check millions of entries and decide which will be the antagonist for CB1, CB2. Okay, so this is the main part of the machine learning, the training of the algorithm, and the other one is the prediction. We need another database in order to carry out the prediction. Okay, so using CB2, I'm going to explain you how machine learning act in looking for novel drugs, so ligands for CB2. Okay. The first part is the data preparations. The data preparations, also for data preparation, we need also an entry. The entry the database we use in this case is PubChem, which is a database from the National Center for Biotechnology Information, which is part of the NIH. And this is how, when we observe the entries in Excel sheet, how the entries look for. We are looking for in this column for these names, in which we can search for antagonists or no-antagonists, we can search for agonists or no-antagonists. So in machine learning, we have only these two entries, yes or not, white or black, 0 or 1. Okay, for example, if we have partial agonists, it could be 0 or it could be 1. For machine learning only is a dual paradigm. Okay, So we have to be very careful in order to give these names for these entries, or 0 or 1. So we need, in this case, we cannot decide at the first time if it is 0 or it is 1. We need to read all the paper in order to decide. Okay, so the training of machine learning required inputs, as I said. These inputs could be 300 of entries, and the training when these entries only say about ligands of the CV1, okay, or about the CV2. They are only ligands. You should be decide if they are agonist, antagonist, 0 or 1. Okay. But for the machine learning, you need to training. So you will be training the algorithm by doing a set of training entries and other set of test training. In this case, 1/5, 1/3, and 1/4 of training test. And you need to use several kernels or integration methods mathematically in order to do these mathematical models. And you need the metrics in order to decide if the machine learning is able or not for prediction. So the metrics are very metrics in machine learning. In this case, the score F1, the accuracy metric, or a C metrics. All of them, they need to be over 0.9, much closer to 1. Much closer to 1, more capabilities of the algorithm in order to predict. Okay, if you have F1 about 0.2, it's the worst algorithm in order to do predictions. Okay. So we need to test or we need to validate our algorithm. Okay, so we need to do benchmarkings in order to validate our algorithms. So it is very it is from a master degree thesis, and it's very boring if I explain you all of these metrics. So I'm going to faster in this part. So we do the benchmarking of all of these kernels, all of these methods, all of these metrics. Okay, so we finally, in one method, we obtain the metrics. Okay, so as you can see, we have for every training asset methods, we carry on. We obtain these values. So these values are our outputs in order to decide which will be the one, the best one. So they are benchmarkings for this supporting vector machine method. These are the validation for random forest method, and these are the validation for deep neural networks methods. Okay, so one of them should be the better algorithm in order to predict. And I want to tell you, we code algorithms for every protein, every receptor. So the algorithm is not the same to any of them. They are code for each one by one, and they should be validated each you are looking for one protein, one receptor. So, okay, this is the part of the training. The last part is the prediction. So we need a database in order to predict, so or a standard database in order to do this prediction is the coconut database. As I tell you, it's about 400 entries. So we are looking for a prediction based on structures, on chemical similarity. So we perform this calculation, which is in reality, which is a matrix of about 400,000 entries with 100 entries for specifically antagonists. And we calculate a coefficient called Tanimoto coefficient. If Tanimoto coefficient is more about 0.5, it is very similar. One is the same, it's identical, okay? 0.5, it's very, very close to the standard molecule, which are the other standard molecules in this case. All of these 150 CV1 antagonists. Okay, with these entries and with the machine learning predictions, our results, our outputs are those molecules, okay, for CV1 receptor as an antagonists. Okay, they are families of flavonoids, they are in lactams, they are isoflavonoids, they are families of isoflavonoids, pyrazolones, etc. Okay, so we have the potentials molecular targets for CV1 predicted by machine learning. Once again, we explore the chemical substructures, the Legos substructures, okay, for finally construct the new structures. But in this case, CV1 has two conformations, the inactive conformation and the active conformations. Okay, the CV1 is, they are the same protein, but they can be in the inactive state or in the active state. But we need to understand as PPARs, the helix H12 should move in order to transduct other genes. In this case, we need to understand if the protein move for the active or the inactive form. Okay. So we study the crystals of these inactive or active forms of CV1, and we we continue with the other part, with the biophysics, the machine learning. I'm sorry, the molecular dynamics. So we choose the active or the inactive. In this case, the active, the inactive, because we are looking for antagonists of CV1. We clean the crystals. We are the docking studies on this protein. And we study with standard molecules as the endogenous antagonist of CV1, the amine molecule, and with the molecule which is crystallizes the protein, AM6, with rimonabant, other molecular study for CV1, or standard molecule which is cannabidiol in this case. Okay. Okay, so, but performing the virtual screenings analysis, we also obtain the binding of these molecules and comparing with the endogenous, which is the level 0, and with energies, repulsive energies in positive and attractive energies of forces in negative values. So we can decide which will be the more stronger for these interactions. And we finally obtain new molecules for CV1, which in this case were the family of the flavonoids. Okay, they are the potential new molecules which can be act as potential ligands for CV1. Also, studies of ADMET, of physical properties, tell us they are very potential ligands for CB1, but with one, in this case, one features. They have many carbons with double bonds, okay? But for an organic chemistry, it is not difficult to change a double bond, no? So the natural molecules, natural products can be modified in a wet lab, okay? But the next point is not always we have the protein, okay? The proteins, they are crystallized, okay, but with ligands that they are synthetic molecules. But this molecule in solid is not the same as in biological in the body, in fluids, okay? But they are a third representation of these proteins, which are the theoreticals. So when we do not have the database of the crystal, we need to construct, we need to build the protein with the theoretical protein in order to do these in silico studies. Okay, so in this case, by doing a match of the active on the inactive forms or the protein CB1 or CB1, we obtain two other structures of the protein. But when I tell you other structure, is not quite different. It's the same, but with the helix moving angstroms, not more. Okay. So obtaining two more proteins for the active, we have three active proteins, and doing the same with the inactive, we have 3 plus 3, we have 6 new proteins, okay? So my space is much better than one crystal. I'm working with 6 proteins, which statistically can be along with the structures of the biological. You understand this point? Okay, bien, good. Okay, the next point is doing the molecular dynamics on these theoretical proteins, and finally, we need to look for which will be the better pose for these new proteins. So we did a mathematical model, which is a dot product. It is quite easy to do this, and we look for which are the molecules much similar as CBD, not CBD, which will be similar to CBD. Okay, doing this computation, these calculations, we obtain these molecules. Okay, arachidine and the DBT, dimethyl biphenyl triol. Okay, but all of them, they are natural molecules. Okay, we carry on with the experiments, theoretical experiments, doing the molecular dynamics in order to analyze the stability of these molecules on the pocket of the CB1, and we obtain stability along the time. In this case, 1,000 nanoseconds, which is very long in computational terms. One of these calculations is about 2-3 months. Okay, one calculation. We need to be we require to do more than one experiment. We need to do at least 3. So 3 months for for one end, and other 3 and 3, 9 months for one curve. Okay, so we need high performance computing to do this. Okay. Okay, the outputs for this analysis is represented in this figure and this table. You can see the new molecules we were obtained by theoretical methods, as I would tell you, machine learning, virtual screening, and molecular dynamics. No? We can see statistically they are different from the crystals. Okay. And we can see that Arachide act differently if they are in the active or in the inactive form. As the same, they are statistically differently if DBT is binding in the active or in the inactive form. And they are also differently by comparing each with each. So Arachidine, in this case, it's preferentially more than the DBT. Okay, so this search of new molecules for the receptors also give us to some molecules which are derivatives of cannabidiol. In this case, for example, cannabigerol. Okay, they are families of cannabis. They are composed from the cannabis, but they are quite similar to cannabidiol, but they are not cannabidiol. And we also obtain some results which are very similar for the binding with CB1. Sorry, and they are the analogs of cannabidiol. And you can see these molecules are very common in our diet. Okay, they are common in carrots, for example. Okay, they are the arachidins. Okay, my second example, and this is very short, is the same with CB2. Not as CB1, which we are looking for antagonists. In CB2, we are looking for agonists. Okay, they are similar, the workflow, but in this case, we have to code other algorithm and validate the other algorithm. So, in this case, we are working with random forest instead of support vector machine. Okay, our entries were not 300. They were they are the validation, and the entries for CB2 were 2,000, more 2,000 molecules. And the potential new molecules in these agonist molecules for CB2 are these molecules, which by doing the LIGO analysis, they must have these features. Okay, finally, my third example and final is this receptor, toll-like receptor 9, which is related with the immunosuppression. Okay, this protein is a dual protein. Okay, which need one molecule in order to put together. Okay, but the protein for human, they are not crystallized. So in the data bank, they are not TLR9 for humans. They are TLR9 for bovine or for horse. And we need to do computational analysis in order to match which could be the structures of the proteins in humans, because the protein is not on the database. Okay, so we do the computations, and these computations give us some results. Okay, these results are the Ramachandran results, in which the black points need to be in the red zone or in the yellow zone, on the brown zone, but not on the white zone. So they can be, these points statistically say to us the black points are on the favorite regions, more about 95%. The black points and the allowed regions in about 5%, and these 2 points, this one and here other one, less than 0.1, they are in these allowed regions. So what means? It means that the construction theoretically by my protein is close than the real one. Okay. The prediction of the protein is quite similar to the protein if this will be crystallized. Okay. And this, and which is the ligand in order to put together 2 of these receptors. Experimental study tell us they are a gene in which mitochondrial gene, which for several reasons I'm not talking about, they generate REN in fractions, okay, and the motifs of these REN, which are related with for binding the TLR proteins, need to have one principal characteristic, which is the C cysteine and G motifs. Okay, so because experimentally they were not characterized the fraction of this REN, we need to propose theoretical fractions of this DNA REN. Okay, okay. Sorry, DNA strain. So we propose 3 models. The model which is composed by 4 nucleotides, the model which is composed by 6 nucleotides, and the model which is composed by 8 nucleotides. But they are all models. Okay. Okay. Doing this, the dynamics studies, the molecular dynamics of these studies, we obtain the binding energies. Okay. So they are the values, and doing a visual inspection of where they are located, these fractions of DNA, we observe they are located in the sites 1 and 2, and according to the literature, for stronger binding and the expression of this protein, it is required that this DNA will be binding in 4 sites. So, which is works in our model. Of course, is this very simple model of the DNA. So we need to construct the second model, the third model, the fourth model. So this is the investigation. Okay. Finally, as an outlook of this presentation, I want to tell you that the natural products are can impact on the new pharmacology, especially with obesity, because obesity also is related with nutrition. Okay. And also there are other opinions, there are other reasons why to use natural products more than synthetic products. Okay, for obesity or for other diseases. But I need to give my final rebuttal with my proper studies. The in silico studies, they are only predictions. They need experimental validations. And there are other concerns. For example, the molecules we are obtaining, we are predicting, not always are in the market. You are not expected to buy on sigma Aldrich the molecule. We need to extract the molecule from one fluid or other. We need to do all the chromatography in order to obtain a product 100% of purity, and so on. Other concerns about this approach are the bioassays. You require to do the validations with experimental. Okay, and the other one, the final one, are the interactions. We are studying one by one receptor with one ligand, but the pharmacology tell us about the drugs interactions, and there is other work. Okay, so they are formulation, clinical trials, etc. So the in silico studies must be taken with they are understand only as prediction and only as a tools. Okay, which is my main goal. My main goal in my laboratory is continue with doing these experiments, not only in using natural database, but also with synthetic molecules, studying the molecular interaction in order to perform a database for finally, I don't know how many years need to complete these pictures. This picture is the main idea is connect the drug target paradigm with the phenotype paradigm, but using the theoretical way. Okay. I want to finally give my acknowledgements for Universidad of Talca. Thank you very much. Universidad de Chile. Thank you very much. Tecnológico de Monterrey, which is my university. My special acknowledgement for my leader, for my chief, Dr. Gerardo Garcia, and the staff of my laboratory, Dr. José Luis Velasco, Dra. Paola Mendoza, Ana Murrieta, Ramón Rodríguez, Priscila, Carla, and Sergio. Thank you also to you for your attention.
Thank you very much.